All right, welcome everyone to the animal movement modeling workshop. Glad everyone's still hanging with us here. It's been a great week. So we've only got a half day here, so we're going to give it as much material as we can in a short amount of time, but we also want to keep it relaxed, like ask questions at any time. If there's any items you guys want to spend more time on, we're totally happy with that, and totally just kind of be a discussion if that's what you guys prefer. Yeah, especially if there's something that we don't spend a lot of time on that you'd like to spend more time on, we're more than happy to. So I'm Brett McClintock. I think we've already met each other this week. And we of course have Devin Johnson here as well. And then we have Mevin Hooten, who couldn't be here, but is joining us remotely. And he's going to start us off with an introduction and then some stuff on point process models. And he may get a little bit into like resource selection, but that's not going to be a big focus today, but we can try and add some time again for resource selection type things if you guys like. So the plan is, intro, point process model stuff, and then we're going to break it up into what's called like discrete time movement models, and then we're going to do more on continuous time movement models. So any questions before we get started? Okay, I believe the bathrooms are just around the corner. We'll take a break, I think, after the discrete time models at the back. So we only have one break planned, but if we need more breaks, just let us know. So with that, Mevin, do you want to take it away?
Yeah, can you hear me okay, first of all?
Oh, is he muted?
Can you hear me okay? Yes?
We cannot hear him.
No?
Yeah.
Can anybody online hear me?
Yeah, I can hear you.
Okay,
Talking
thanks.
now?
Yeah, we can't hear you now.
Okay.
They can hear him online. Yes.
Oh, they can?
Yep, it sounds like they
Okay,
can.
we can't.
And are you seeing my slides also?
Okay, try again.
How about now? Can you hear me? Anything? No? Sounds like I can hear the room okay.
Okay, there we go.
Okay, better. Great. Hey, can you see my slides okay?
Yeah.
Okay. Looks like my video froze, but that's okay. Let me try those again. We're having some issues. Okay. 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 You should be seeing the title slide. Is that right? Yes. Okay.
Yes.
I see some nods. Okay. Great to see everybody. I'm Mevin Hooten, and as Brett said, and so it's great to see everyone. I'm sorry I couldn't be there. I've got we're in our final exams week here, so things are crazy, but I'm really happy to spend some time with you today. Let me give you a couple sort of segments here to start things off today. It'll take about 45 minutes. I'll do a little bit of an intro to this topic. And of course, many of you are already familiar with this topic, and that's why you're here. And you've had some great talks on this subject this week. So you're already primed for some of this info. I think my big overarching purpose this morning is just, or for me this morning, for you this afternoon, is to get you up to speed on kind of what this field is about, and then give you a bit of the background about how relatively simple it is to get started in this field if you haven't done anything before. So we're talking about animal movement. Before I get there, I just wanted to say thanks to everybody who organized the meeting this year. Again, sorry I couldn't be there, but I've been to this meeting before and I love it. And I know a lot of folks work really hard to put this on. So thanks so much for hosting it, those of you, and Evan for doing all the web related stuff too. So here's what we're talking about, the statistical analysis of telemetry data. That's the key here. So statistics is important, the data portion is important, and we'd like to understand how to use these sorts of data to better understand the life history and ecology of animals. Most of us know that even though Euring is primarily a bird meeting, it's sort of branching out into all sorts of other species. And we know that there's a variety of different ways to collect what I call telemetry data. And when we say that, it could be a variety of different types of data, but for the most part today, we'll focus on the type of telemetry data associated with recording or the observation of positions or locations of individuals over time. And so we can do that with a variety of these devices. For example, there's neck collars, GPS collars associated with large carnivores. We have similar neck bands associated with birds. So this is for the Euring crowd, it's greater white-fronted goose. And the idea is then what sort of data these collect and then how we can best understand them. Well, in the early days, this is what we would get from 4 greater white-fronted geese flying from Ireland to all the way to Greenland, stopping over in Iceland. And we would like or seek to use these data. These are, you can see these points on this map and different colors associated with different individuals. We'd like to use these data to better understand where these things are going and maybe the timing of when they do what they do during migrations and other life history things that are important for the species. And so in the early days of animal movement modeling, I think one of the big challenges was how we do that rigorously, statistically, how we sort of connect the dots in a way. And so here, using some continuous time modeling approaches, here's sort of a reconstruction of where we think these birds may have gone during this period of time. And then some understanding of, let's see on the bottom here, this is in generalities, of course, but on the bottom here in terms of like when the migration might have occurred for these individuals based on our observations of their positions over time. So that's kind of where this all started. In the modern era, statistically, was how we use those data to better understand where things go. Then people started asking why they go where they go and what decisions are they making. How does that tie in with their physiology? How does it tie in with their conspecifics and allospecifics? And then, you know, at the population level, what are we seeing? And so I'll get to some of that here in a second. Even though I said in the modern era, this concept of animal movement and trying to understand how it works is old. It's really old, right? This is Aristotle talking about it thousands of years ago, and we're still talking about it today and trying to understand how to do things with these data. So one of the things we did in this textbook on the subject is try to, and this is sort of the purpose, I think, of writing it, at least for me, was to try to put together these concepts in a familiar framework, one, and I'll talk about that in a second, and then also in a consistent statistical notation. So one of the nice things about this book is that we sort of framed all the models in the same notation. So we use the same notation for observed positions, S, we use the same notation for latent positions, the true, unknown, usually latent positions in these statistical models as mu, and then regression coefficients are always beta. We use the same consistent notation for matrices and vectors and all that. So it makes it kind of easier to put all these things together and try to understand how they're similar and different. When we went through the literature on all this to put this book together, at least I, being a spatial statistician, sort of started to figure out that they talk about these models in the same general framework that they talk about spatial statistical models, in the sense that there's point processes, discrete time processes, and continuous time processes. And in the spatial world, that would be discrete space and continuous space. And so we decided to break it up in the book in that way. And that's how we'll talk about it today as well. And we'll do it in this order today. So I'll give you a feel for that here in the next few slides. But the point process approach, and let me help you out with some of the notation here, is essentially like this. Suppose that we have some understanding of these positions. Let's call them mu. They're bold because they're vectors, they're coordinates in space, latitude and longitude, or whatever coordinate system you're using. And over here in the picture on the right, you see those black points, and those black points correspond to the positions that the telemetry device is giving us. So this is kind of one of the original ways to analyze these data, using what was referred to as resource selection functions. But it wasn't until after a while later that people realized these are really just statistical point process models, and so we can use the same framework for that. The square bracket notation here, so mu arises statistically, stochastically from some distribution. This is a distribution of interest, so it's just a shorthand notation for that. And this distribution could be characterized by the product of two functions. One, f usually is referred to as availability, and g is referred to as selection. And I'll talk to you about that in the next section here when I switch the slides up. That would be point process models. So the positions themselves are considered as points in space, and they're considered to be the random variable that's observed. The discrete time modeling approach is, at least in my mind, is better understood from more of a classical time series perspective, where you would have discrete snapshots in time that you collect data, or you're interested in the underlying process, and the evolution or dynamics of that process over time is what's important. And so one way to write that is as a time series model. And in this case, you see down here, we're using a V notation for velocity. The velocity would be like speed and turning angle together. And then a position, the location in space, mu, could be like adding up all those steps in certain directions. And that's one way to think about these things. Another way to think about them is the way that Brett will tell you about when we get to that section, using the same ideas, but in polar coordinates, where you're focusing more on directions and steps. Okay, that's discrete time models. Continuous time models, just as a big overview here, Devin will talk about later, and that is effectively the same idea, thinking about time series in a way, but thinking about it now in continuous time, such that there's infinitely many small steps you could think of in a way that accumulate to create the trajectory of interest. And understanding the dynamics of how all those small steps happen is really what the continuous time approach is about. And usually that corresponds pretty well with what modeling approaches are used in stochastic differential equations and those sorts of things. And so while it sounds a bit more heavy quantitatively, it's really just sort of a few fundamental ideas that you can start to see how things work. And we tried to cover that in a way that's sort of a natural progression of those ideas in the book. And then there's a number of great papers on the subject too. And so Devin will show you that. One of the cool things about these approaches is, let me go back two slides. The point process models, I'll show you in just a second how simple it is to implement these things on your own. While there are also several software packages that can fit these models to data, you don't really need any special software. You can do it with almost any statistical software. And I'll literally show you that on a few slides in R, how you can do that. So you don't need any of the custom software, but some of them might make it easier to organize your data and things like that. The biggest hurdle there is preparing the data. The discrete time models, Brett has a really nice software that he's worked on quite a bit, and there's other ones he'll discuss as well related to that for fitting those models. And so they're really straightforward to do, but you can also do this on your own. Let's say if you want to do it in a Bayesian framework, you could use JAGS or Nimble or something like that. And then the continuous time models, Devin has a great piece of software that's been around now, what seems like for ages. And he'll tell you about that. Is everything still going okay? Can you all hear me all right?
Yeah. I have
Okay,
a quick
yeah.
question.
We have a question.
I was just gonna ask, are you gonna be able to give us these slides, or should we take notes?
Yeah. Yeah, I think Brett, can we share we have this on a Google Drive. Can we share that, or should we just?
Yeah,
Should
yeah,
we
I have
just-
everyone's email address, so I can just share that whole folder with everybody.
Yeah. Yeah, so they're all yours, and there's some code in there too. So you can run some things.
Well, thank you.
And sorry to move a little quick. I want to make sure that Brett and Devin have plenty of time today to show you stuff in live action. Okay, what can we do with all this? So these are the three basic approaches: point process, discrete time, continuous time models, right? What can we do with it? Well, you can do things like couple it with physiology if you want. And so there's a lot of things you can build on now with these basic ideas. And I'm not saying we're going to cover these things today, but this is just to give you some hints on like where you could go with these ideas. You can start trying to couple it with decision-making processes that are tied to physiology of animals. And we talked about that in this paper on recharge dynamics. Another thing you can do is sort of ask, how would you get hierarchical or population level inference using hierarchical implementations of these individual level movement models? That's a common question. If we want to make inference on individuals, we have a variety of ways to do that. But how will we scale that up to learn about what populations are doing in general and what's the variability associated with that is important too. And there's a variety of good papers on that too. There's natural hierarchical frameworks. You can embed all these in, and variety of computational techniques you can use there too. Here's some examples. And then finally, one thing I want to make sure to mention is that a recent thing, and this really couples nicely, I think, with the Euring concept, is that a recent movement in this literature is to sort of couple models for telemetry data with models for capture-recapture data. So capture-recapture data is also individual based, right? And the idea is if we understand how things are moving around in the space, where we're also observing whether they're present or absent in certain places where we're recording them, we can couple those things explicitly. And there's a lot of great progress in this area. It's not my area, but it is an area that Brett works and Devin work in. And so hopefully if you have questions about some of that, I assume there were a few talks on that this week too. You can chat with them about it later, but I wanted to make sure there was a special issue or a section of an issue in ecology on this recently, if you want to read a few papers about the modern approaches to dealing with telemetry data embedded in spatial capture-recapture models. So this is just barely scraping the surface or scratching the surface of what's available out there now. This field of animal movement has really gone crazy in the recent years, and it's been exciting to watch the progress there. I think what I'll do now is I'll switch over to point process models, and we'll go a little bit more in depth in that topic, and then we'll switch it over to discrete time and then continuous time for the rest of the afternoon. Is that okay with everybody? Okay. Thanks for the hand gestures. That's the only way I can tell if people are able to.
Thanks, and I forgot to address the folks in the chat earlier. If they do have questions, they can just post them and we'll periodically monitor them. And if anyone on a computer notices we've missed a chat, please let us know so we can address that.
Yeah, I was going to say that too. I'm glad you mentioned that. I can't see them very easily, so so just stop me if anybody wants to stop and chat about anything. Okay, so now that we covered the overview of like what this field is about, there you can imagine there's a variety of softwares, a variety of approaches to do it. What I wanted to do here to start us off is sort of demystify some of the simplest thing, the oldest things that have been done in this field, and tie this in with resource selection, as Brett said, and give you a feel for what the step selection is all about and why people call it these names. And I'll do my best to do that and sort of hopefully disarm this topic a little bit, because it's honestly not very complicated. There's one or two basic fundamental concepts to get across, and then I'll show you computationally. It's almost like a one-liner in a statistical software to do it. So that's where we're going here. Okay. So let's go back. Let's return to this concept of a point process model and what it is. So remember, here there's a position, let's say S. This is, again, a coordinate in space, the space of interest. S that's indexed by I for telemetry fix I. So imagine that you had n telemetry fixes In other words, a measurement or a recording, an observation of where the individual was of interest over some period of time. For now, we assume that these things are conditionally independent, although they don't have to be. If they're conditionally independent, you might write a model, statistical model for them generically like this on the top. It's this simple, just create a probability distribution associated with the random variable being the point in space. It depends on some underlying parameters beta. And then the script S is the study area. So in this case, it's this region that's outlined here effectively with this grid that you see in the background. Okay, the difference between that and what's called a spatio-temporal point process is that there's some dynamic there to account for the temporal part. So now we're not necessarily considering the fixes to be conditionally independent. Here we're considering them to be dependent in time, and that dependence is important due to the dynamics of how the animal moves and makes decisions as it's moving through its landscape or waterscape or whatever it is. And so here we have S that's denoted as a position associated with time, T, and fix I. So now you could imagine the time becomes important, and you could somehow build a model that conditions that position on what may have happened in the past. This would be a simple sort of autoregressive perspective on that model, but there's a variety of other approaches you could use there. And everything else is basically the same. There's still a set of fixes that you would collect, and the positions correspond to some geographic space of interest. So this is a generics of what a point process model looks like. The only thing we have to do is we have to sort of specify what these distributions look like. And the cool thing is we can do that somewhat generically. In the like traditional resource selection world, you could use a weighted distribution form of a point process model that looks like this. So now we're defining the probability distribution for the random vector S as some function G, that's called the resource selection function, over the integral of that function in space. So just integrate that function in space so that G integrates to 1 over the spatial domain of interest. And that just makes it a valid probability distribution. So you can pick any positive or non-negative function G that you want. Most people use exponential form of this. So it's E raised to the X times beta. So it's got a regression looking thing, X times beta in there that controls the intensity associated with where, or density, if you like to think of it that way, associated with where the points might occur. And the goal then would be to estimate and learn beta, and then make inference about beta as in that case, you can only make inference as relative selection. So in that case, you would say here, if beta is really large, then this particular covariate suggests that model suggests that the individual is selecting for that covariate more. Okay, so don't let this, for those folks who aren't, oh, go ahead. Yeah, sorry.
Sorry, what is the role of X?
Yeah, right. Thank you. I didn't describe that very clearly. So X is a set of environmental variables, usually, that you think may be influencing where individuals decide to spend their time. So things related to habitat are obvious choices, but a lot of times people use GIS-derived variables like elevation and slope and aspect and percent forest cover and other things. So you can think about X as like in a regression setting, it'd be like your design matrix, the variables associated with predicting the response. So E to the X beta is what we're looking at here for G in the most common setting. And fitting these models can be done in a variety of ways. Here's 3 approaches. 1, you notice that has an integral in it. That integral is often impossible to calculate by hand, but it's not terribly hard to calculate it numerically. It's just that it might not be the most efficient way to do that numerically, although I've found that it's not too bad to handle. However, there's a couple of other approaches you can use to sort of bypass direct calculation of that integral in the bottom. Notice you can't do anything with this likelihood. This is like a likelihood in a way. You take the product over I here of this fraction. You can't do anything with it directly unless you can calculate that integral. So you can't maximize the likelihood or use it in a Bayesian context or any of that until you calculate that integral. And you might have to do that repeatedly. So that could be a crux in fitting these models. That's the challenging thing about point process models. So how does it work? Well, like I said, you could numerically do this integration, which is not too bad usually, but maybe not always intuitive, and it requires some custom code. Or you could use one of these two approaches, either Poisson regression or logistic regression. In the logistic regression case, you would take a set of background points from the spatial domain of interest. You'd call those things zeros, and then you'd call the positions where you observe the individual ones, and then you trick an algorithm into fitting a logistic regression to those zeros and ones. As long as the background point, the number of background points increases, you will obtain the same inference for your coefficients beta, it's a selection coefficients, as you would if you did the integral directly. So that's a great trick. And the same thing is true of Poisson regression. Instead of, in the Poisson regression case, you just grid the area up into grid cells and count the number of points that fall in each grid cell and use those as your data and fit a Poisson regression. And this can be done, and I'll show you in just a second, trivially in R. It's mostly about just the processing of the data. That's the hard part. So let me show you how that works. First, you define your study area. In this case, these are sort of home range based study areas, but you don't have to do it this way. You could define a larger study area. You just have to decide where you feel that your inference should be restricted to. This is using a KDE sort of isopleth, so we don't want to go into details here because this isn't a home range analysis workshop, but here's a KDE with a relatively small isopleth. Here's one with a larger one. I think this is 90%. This is 95%, or I think this is 99% actually right here, this C. And then this would be a convex hull if you wanted to use that. There's a variety of approaches there. It may change the inference slightly because the study area is changing, but that's your call, your decision as an analyst when you fit your models to these data, depending on the sort of inference you want. So let's use this C here just for an example, and I'll show you how it works. If you take the logistic regression approach, you'd take our original data here on the left, and you'd couple that with a larger data set that's taken randomly from that same spatial domain. You call all these zeros, and you call these ones, and you fit a logistic regression. Associated with each one of these points, the covariates, the x's, would be measured characteristics of the landscape that are associated with each position in space, both the augmented ones here on the right and the observed ones here on the left. And then you literally just fit a logistic regression. You take some covariates. Here's an elevation covariate, an aspect, and a slope, or let's say slope here is B and aspect is C. This is just as examples. These data, by the way, are from a mountain lion in Colorado in the US. The other approach is a Poisson regression approach. And here you'd have your original set of data points on the map here on the left. And then those are just binned up into these grid cells here on the right, where we just simply count the number of points that fall in each grid cell on the right. And then your data are a bunch of zeros for all these grid cells that are white. And then for the grid cells that are gray or slightly darker colors, black, those would be higher counts. And we could model this with a Poisson regression instead. It's all the same thing. It's all the same thing asymptotically. As the grid cells get small, or as the background sample size gets large, in this case, you can do this in these ways and they'll give you the same results. And I'll show that to you now. In R, it's literally as simple as after the data are prepared, using this one line of code. So here's the important part in R. For those of you who are familiar with R, the GLM function will fit a generalized linear model. This is the Poisson version. The Y is our response variable here. That's the grid cell counts. Elevation, slope, and exposure. So exposure is like aspect just pointed in the southwest direction in the northern hemisphere. And those would be my covariates. I just tell it I want to fit Poisson regression and use a log link. I'll give it the data set that I've prepared ahead of time, and you'll see all the code for this in the files that Brett will send everyone. And let's look at the results. This is the output in R. The only ones you want to make inference on are the ones associated, the coefficients associated with the covariates. And so negative 0.29, negative 0.21, or sorry, 0.21, and then negative 0.13 are our coefficients. So in this case, it's selecting against elevation, but for slope in this case, so steeper slopes, and then against exposure, so less exposed aspects. And that's how you would make inference here for this particular individual. Now, notice that over here, these p-values are associated with, just like you would do in regression, the strength of those effects. So really, the only ones that are important here are elevation and slope. We're not seeing conditionally on the other variables in the model, we're not seeing a strong influence of exposure on resource selection for this individual. That's it. It's one line of code. So this is why I said you don't really need custom software to do this. Now, this is using the Poisson perspective, but you don't have to do it that way. You could do it with the Bernoulli. So this is the logistic regression approach, this exact same problem, just the data are set up a little bit differently. So you notice I changed the data set name there just to say, okay, now these response variables are zeros and ones, and you still have covariates associated with each one of those zeros and ones. And we're going to fit the model using logistic regression. And now look down here at the coefficients again for the three variables, negative 0.3, negative 0.2, negative 0.1. Let me just cycle back and forth here between the Poisson and the Bernoulli. They're a little different at the second decimal place, but other than that, you'd get effectively the same inference out of it. Now, why are they a little different? Well, again, it's asymptotically, right? So if your background sample gets really large and your number of grid cells also gets really large, or the grid cell size gets really small, they will converge.
One question, Mevin.
Yep.
Would there be an advantage to do conditional logistic regression without the intercept?
Yeah, so here you could do that, conditional logistic regression, and I'll talk about that in particular on the next few slides. And so maybe it's a good time to transition to this next topic, just so that we're not belaboring this RSF type of stuff, but this will give you the correct inference here. So let's extend this one level up then, so we get to that question and build it into the rest of the topic here. How do you deal with it? The concept of your telemetry positions are close in time, and now you've got positions that depend on each other, like where you were at the last time is also important relative to what you're selecting in terms of resources at the current time, right? How do you deal with that situation? Well, this is where the spatio-temporal point process idea comes into play. And so here you would use a similar setup where you've got your data S, these are positions in space, these are coordinates again, this is a vector, lat and long, associated with some particular time, and it just arises from some probability distribution that we need to specify that I say generically looks like this. So we just need to define this. Then we can create a likelihood using that. So here's one way to define it. And this is the most common way, probably, using that weighted distribution idea again. Notice that it has a similar form as before. It's a fractional form where you've got now a product of two functions in the numerator, g and f, and then an integration of those two functions in the denominator with respect to the space, the geographic space. That's it. So how does it work? What are the two functions? Well, here, x is the same as before, and f is the new part. So f is what's referred to as the availability function. So it's, in other words, you can think about it as how the dynamics of the movement trajectory works on its own. And then g would be the selection function. So g could be the same g function we used before. Like, for example, you could use e raised to the x beta. That's the most common thing to do, although you could use other ones too. And then f, we have to decide what to do with that. There was an f that was hidden in the previous one. Let me show you where it was. It was hidden there. G times f. Well, here f is 1. And so it's like saying f is still there. It's a uniform on the space of interest, S. So uniform distribution on the space of interest. So there is an f there. It's just sort of implicit. So now let's go forward and let's think about the space-time version of this. Now F can be anything you want, but it makes sense if it's something related to the movement, the actual dynamic component of the movement related to the trajectory of the animal. So you can imagine now it gets complicated because you could think about all sorts of different like movement models in a way, and Devin and Brett will talk to you about some of those in what follows. The simplest possible thing you could do is you could just define F as still a uniform distribution, but it's centered on the previous position in time. So uniform centered on that position, and it's uniform in the space, so you have to define the boundary for it. So you could make it a disk or a circle that's centered on the previous position, and you could say, okay, the diameter of that disk, or let's say the radius, is based on something related to how far we think the individual moves in that amount of time that's elapsed. And that's probably the simplest way to do this and what's pretty common. So let me show you how that works. We would fit that model using the same, one of three of the same approaches. You could do numerical integration, or you could use this logistic regression approach or Poisson regression approach. You could do this in either of these ways, and some softwares exist to sort of aid you in doing this. And I want to make a point here. It is a little bit different in the sense that the model now, if you were going to use logistic or Poisson regression, it's very similar. The only difference is that you have a step-specific intercept. I think this is very little known. This is little known. This is not something people talk about. But the correct statistical model is, let's say, logistic regression. I can't remember how I think I do it with Poisson regression. Let's see. I'm going to give you a demonstration of that. So here's our three covariates. This is a different mountain lion, but where the data are collected sufficiently close in time so that you can see there's a dynamic movement trajectory happening as the animal moves around in this large circle. This is again in Colorado. This is a elevation covariate. This one is a slope covariate and an aspect or an exposure covariate, similar to last time, but now we're looking at finer scale decision-making and resource selection. By the way, because these are broken up into little steps, we would call this a step selection function model. What are the steps and how do we accommodate that in the availability function? Well, we've got these now associated with our trajectory that you can see as that line under there with these little points. Now you've got these big circles drawn around there. These are the availability distributions. They're step specific. So at each point of interest, what was available to you starts at the point before you and then some buffer region around there. Now you can imagine there's a variety of ways to generalize this idea. You don't have to use circles. You could think about it as being a tapering distribution that tapers off from the ground samples now because we have one for each circle. And then the position that we actually observed it moving to would be the one. All the rest of the data would be zeros. That would be the logistic regression. But you can imagine every circle moves to a new position, right? So you kind of start over and you want to do this all together holistically. And so the way the model is set up that you'll use to approximate the point process distribution is as a logistic regression, but with step-specific intercepts. So let me show you what I mean by that. So Yij would be for step I, J would be, if J is a 1, it would be my actual observed telemetry position. That would be a 1. All the other Yijs for all the other Js would be zeros. They would have Bernoulli probability P. That would be, this should say logit here instead of log, but anyway, that should say logit. I can fix that on the slides. They would have an intercept beta naught at step I. And other than that, they have their covariates X and then their coefficients beta. That's it. You're just fitting logistic regression again, but with step varying intercepts. So that will get you really close to the correct inference, approximately close. And I've noted, my experience with this is that it's imperceptibly close in most cases. Now, where does this comment that we had earlier come in about conditional logistic? Well, it turns out because you only have a single one in this position and here, and the rest of them are zeros for each step, you should condition on that. That's a constraint. You can only have a single one and the rest of them are zeros. And so instead of this likelihood is what you'd use to fit this model. And like I said, usually this is pretty close regardless. Instead of this directly, you could use what's called conditional logistic regression. And the way that works is it conditions on this constraint that the yij's for each step I sum to 1, because you only have a single one in there and a bunch of zeros. And so you end up with a different likelihood component that looks like this. It actually simplifies quite a bit relative to the one I showed you before. The only difference is it doesn't have the intercepts in it at all, because they'll all cancel out. So what's nice about that, we don't make inference on the intercept anyway. You make inference on the coefficients associated with the covariates. What's nice about that is it doesn't waste any extra time, and there's built-in software for doing this in R. And once again, it's a one-liner. So here, if you wanted to do it using the GLM approach I talked about before, where you have step-specific intercepts, then you would use the GLM function with the Poisson over here. You'd have a bunch of intercepts out here in the output. You can ignore those. You go down here to the coefficients and you say, okay, these are the coefficients associated with my covariates, elevation, slope, and aspect. And you notice real similar coefficients as what we saw last time. And you could make inference on those directly using this approach. So negative 0.41 would be negative selection about elevation. 0.09 would be slightly positive selection toward slope. Negative 0.5 would be negative selection toward exposure. What does the conditional logistic regression amount to? Well, again, it's one line in R. It's literally one line. After you have your data organized, it's just one line. So this is great. Here, you just look at these coefficients and you notice they're the same. Negative 0.4, 0.09, negative 0.5. So what most people do when they do step selection analysis is they'll use the conditional logistic regression approach. That is the proper approach for doing it. You don't have to. If you like to build this into another approach and deal with all those intercepts, you can. But this is pretty clean. This is non-Bayesian, right? This is just maximum likelihood based. And it uses, you notice it's saying it's doing something with Cox proportional hazards model. That's because conditional logistic regression is often used in survival analysis and other sort of things. But we're tricking it here to use it to do step selection function inference for us. And that's it, though. You notice it's a regression type statement. It's just that you have to tell it right here which step you're on. It's kind of like putting that intercept in there in a way, but it's just sort of telling the software that does conditional logistic regression which step is which. And that's it. You get the same inference out of it either way. So I hope what this sort of demonstrates is that you shouldn't be too afraid of these methods. The point process models themselves get kind of complicated if you want to really get into the mathematics of how they work. But in terms of how they're used for animal movement, most animal movement inference, they're pretty straightforward, both from a implementation perspective and then computation themselves. It's literally, you saw it's sort of one line here in R. There are some great packages by friends of ours. AMT is a good package that is developed by John Feeberg, and Tal Avgar is involved in it. And gosh, who's the other person, Dev?
Signer.
Oh, yeah.
Johan.
Yeah, Johan. Yeah. And they've got a nice software. I haven't personally used it because it's literally only one line in R to do most of this stuff. But if you want to, it's there, and there's great documentation on it and great papers about it too. It does essentially the same stuff. I think I'm out of time.
Okay, well, this would be a good time for questions for Mevin. And Mevin, are you going to be sticking around? Is that right? For at least
Yeah,
a
I'll
while?
hang out. Yeah, I'm gonna
Okay.
hang out with y'all. Yeah.
So do we have any questions before moving on to discrete time movement models?
We don't discuss position uncertainty here, so it's impossible to account for those in these models. If we're uncertain about positions a little bit in different ways.
Yeah, great question. Let's go back to the model itself. Well, let me go to the even simpler one here. Let's go to the point process one just for a sec. Okay, so you notice that the question was about uncertainty in the positions. So here, we're not assuming that there is any uncertainty associated with positions, but it's not challenging to accommodate that. Here, all you have to do is build a model hierarchically that has another source of error associated with it, the telemetry error. So now you could imagine that instead of it being S arises directly from a point process model, S arises directly from a model that depends on an underlying latent true position mu, and then mu is the variable that mu being a coordinate set of coordinates associated with the true position comes from a point process model. So it's not terribly hard to do that in a hierarchical modeling framework. Now, the catch is it's not a one-liner in R then. But it's not impossible to do. That's a very straightforward thing to do.
Yeah, and I think in sort of response to that, because a lot of these models that we develop here are going to assume that we know the actual location of the animal. And so what we tend to do in that case, and I think you're going to talk directly on it, right? The imputation. Some of the models I'm going to show later on are involved in this, is that you have sort of a stochastic method where you simulate what the true location might have been given the data you saw. Then you fit a model, kind of store the results, and then repeat it often, and then kind of look at the ensemble of results that you got from that. And so, yeah, we'll talk about that later. It's kind of a trick to get around that. Now it's no longer a one-liner in our problem.
Is there any, I'm curious about this logistic regression trick for estimating the betas. Is there any kind of intuition in why that's, yeah, I don't understand why this works. Is it obvious? Am I missing something obvious? Or?
Yeah, here's the intuition. What we have to do basically is we have to sort of approximate this integral in the denominator here. Eventually, if you multiply all these things together for all I, you get likelihood of interest. The cool thing is the logistic regression approach can be shown. There's a couple good papers on this, one by Garrett Artz and one by David Wharton, that show that these things converge when you have these logistic regression implementations under cases where the sample size for the background sample increases. So you get the same basic inference out of the logistic regression. The likelihood approaches the correct likelihood based on the point process model.
The zeros are kind of approximating that integral by introducing all those zeros. You're getting at that integral and then in the denominator. Does that make sense?
I'll have to think about it.
Well, don't think too hard about it, because it's not like a super trivial proof of that, but they do describe it in the appendices of those papers, like how it actually works mathematically. So it's worth taking a look at that.
Yeah, that'd be.
So maybe you said that the beta coefficients are not easily interpretable, except for the plus and the minus, but maybe you can say something about how you would discuss or present the coefficients, whether you would make some odds ratio or something to like an effect plot or.
Yeah, it's relative inference. Here's the problem. So I'm glad you brought that up. There's a great paper by John Feeberg about this. If you want to read about more on resource selection inference and interpreting these things without going into too much detail on it here, that paper is really good. So check that one out. It's fairly recent. I can't remember the journal, but it's not challenging to find on Google. The short version is, notice that if you plug in as G down here, the selection function, E raised to the X beta, then if you happen to have an intercept in there, beta naught, it cancels in the numerator and the denominator. So you can't have an intercept in this, which means you can't get at exact probability of selection. You can only get at how it affects relatively the chance that a point is going to occur at the particular position in space. So that's why we're sort of limited in the inference. However, that's only using the standard approach G as E raised to the X beta. If you use a different function there, like for example, a probability-based function, like instead of E raised to the X beta, you did something like logit or probit raised or of X beta, then you can get inference on actual probability directly. And there's good papers about that too. And it's not always intuitive, but there's various ways to connect that with other sorts of dynamic movement models. And we have a good paper on that where we talk about that. And then it leads to some really weird looking G functions here for selection, but you can use those if you're more interested in inference that's more specific.
Any more questions on the introductory? Oh, here we go.
Regarding the step selection functions, and you were mentioning the step-specific intercept. I had kind of expected that this would translate into a random effect, maybe on
Oh yeah,
those step ID, but I did not see that exactly. So is that also a possibility, or is that not? Doesn't it make sense?
yeah, that's very observant of you. Yeah, when you see something like this, beta naught I, you naturally want to say, oh, that should be a random effect, and it should come from a distribution whose parameters are estimated. Yeah, very observant. In fact, there are papers about that. But I think the point here is it's not necessary here. You'll get the inference correct either way. And what most people would do instead is just use the correct conditional likelihood that conditions on this this constraint that at each step the data sum to 1. So what I would do if I wanted the exact inference, and it's a little more efficient too, is just fit the model with this likelihood instead. So we can play games if there's a reason why you really want that random effect in there, and you want to keep the intercepts because it couples nicely with how you're building this model into another one or something more complicated, then I think that's a reasonable solution. Otherwise, it's not necessary.
Okay, well, if there's no other questions, I think we can move on to the discrete time portion here. Is there any questions in the chat?
I didn't see any.
Okay.
Yeah, if anybody has questions in the chat, put them in there.
We should probably warn our people in the chat that I tend to walk around, so if you're just getting my butt, let me know and I can move out of the way.
Which one are you doing?
The discrete software. So now we're going to move on to what we call discrete time movement models. And there's different flavors of these, but for this kind of shorter workshop, I thought it might be helpful to go over some of the more popular ones that we've been seeing popping up at meetings like Euring or ISAC, if anyone attends that one. And these involve hidden Markov models. And oh, thank you. And one of the main ideas behind these is that, here's like our huge data set. It's hard to tell what's going on when you just look at a figure like that. But what they enable is inferences like this, where we can actually break up the track into what we think are different behaviors. In this case, these are southern elephant seals, and this helps identify where we think they are foraging, right? We don't know they're foraging here, but that's where we think they are. And that may seem trivial because you could think maybe you could just look at these tracks and you can identify where the little spaghetti plate is, and that's where they're foraging, but it's not always that easy. And it's also a very quick way to do it. You're not doing it by hand, right? So I'll give a little background on these types of hidden Markov models first, and then I'll go over 2 R packages that are designed to make this easier to do. And they provide ways of processing your data and then fitting the models and then checking the models. So first, for some background here, one of the most basic movement models there is the correlated random walk. You have these in both discrete and continuous time. Like Devin will talk more about the continuous time version. But the idea, it's pretty simple. On the left here, we have what's called a simple random walk, where all that determines your next position is your previous position. And then there's some sort of scale parameter. So you just bounce around randomly from your previous location. But in a correlated random walk, there's directional persistence in your movement. And this can be a very handy one for animals. It's very simple, but it captures the fact that there's a persistence in movement, especially if they're going after a target or something like that. So in these discrete time models, you don't have to do this, but what has become very popular is instead of modeling the positions that are themselves through time, which we're going to call X right now, you chop up the tracks into step lengths, which, as you can probably guess, is just the length from between our points, right? And then there's also a turning angle, which is a measure of the change in direction between 3 points, though, okay? So it's measuring the tortuosity in our track. And we can assume just different distributions for the step lengths and turn angles, okay? So our step lengths have to be some non-negative distribution. One that's often used is the gamma distribution, and these are just some examples of different gamma distributions. And then for turning angle, we have to use what's called a circular distribution. And a very popular one is the von Mises, but there's other ones like the Rapp-Cauchy. But this is bound, on the circle, basically. So the one thing that's to note here is that if our turning angle, if you remember, it's the change in direction. If that's concentrated near 0, that means the animal is maintaining directional persistence. That's going the same direction that it previously was, right? And then if our turning angle is focused more around the edges, you're at pi or negative pi, that means the animal is switching direction, right? So these can be handy distributions for when we get into the actual modeling. So yeah. But the idea behind these multi-state, yes.
So if one had a uniform distribution on the turning angles and a half normal on the step lengths, would that be the same as a correlate? I mean, as a simple random walk?
Yes, exactly.
Okay.
Yeah. So the idea in these multi-state random walks or these hidden Markov models is that the animal can switch between different movement processes, and these will correspond to proxies for different behaviors. Okay. And what they key on is that this behavioral process is an unobserved Markov chain. So in a first order Markov chain, your behavior at time t only depends on your behavior at t minus 1. Okay. So it only remembers back one time step. But it's this unobserved Markov chain. And if we just think of an example, we have a very simple example. We have 2 states. The animal can either be foraging or transiting between foraging patches. This is governed by the state transition probabilities gamma. So we have a probability of transitioning from forage to transit. We're calling that gamma 1. And then the probability of staying in our foraging state is then just 1 minus gamma 1, right? And then similarly for the transit, we can either go from transit to forage with probability gamma 2, or we can stay in transit with probability 1 minus gamma 2. Okay, so that's the unobserved Markov chain that we're modeling. But the problem here is that we don't observe these states, right? We only observe these step lengths and these turn angles. So the hidden Markov model allows us to try and infer what the underlying latent states are, which is what we're really interested in learning about. So through time, we have this underlying state process. In this case, we're thinking of transit and forage. So it's proceeding through that in a Markov fashion. And then we only observe though the step length and the turn angle. So what we want to do is learn about these unobserved hidden states based on our imperfect observations, being the step length and turn angle. So this hopefully sounds familiar to this crowd because, you know, pretty much every model that we talk about at Euring can be formulated as a hidden Markov model. So hopefully pretty familiar, like all the classic capture, recapture, occupancy models are all hidden Markov models. They can all be expressed this way. Yeah. So for an example, we could assume that our step lengths L follow a gamma distribution. And the key here is just that we make the parameters of that gamma distribution state dependent. That's what that subscript J there is, right? So we just switch between these different gamma and von Mises distributions depending on the state. Any questions so far? Okay, now we can get some more biology in there by trying to incorporate things like covariates and how they may affect that state transition process. So, you know, this here is just saying, does insert covariate have an effect on the probability of insert behavior? So you could take anything you want, like things like slope or elevation or habitat type, anything you want. And you can make these state transitions a function of those covariates just using a simple logit link, right? So that's where you can start putting interesting hypotheses into your model related to behavior. So I'll now go into some examples using package MoveHMM. There is going to be, since this is a workshop, but it's a very short workshop, there's going to be code and stuff up here, but it's all available in the package vignette. And similarly for the other package momentum that I talk about, every bit of code is in the vignette. So you guys can work through all those examples if you're so inclined. So just a little bit of background on the package of MoveHMM. The lead developer is Teo Michilo. He's actually now at Dalhousie University. But the package is available on CRAN. And as I mentioned before, a great place to start with it is the vignette. Teo put a lot of work into that vignette. And there's also a lot of just good documentation for the package. And there is a reference in methods in ecology and evolution, but that's fairly short. The vignette, and he's got several vignettes now. They're very, very detailed and are a great place to start. So a little bit into the workflow of the model, of the package, sorry. It has functions available for visualizing your data, and then it has a very flexible structure for specifying the type of model you want to fit. You can specify the number of states that you want. You can have different distributions for the steps and the turns, and you can include covariates into your state transition probabilities. It then fits the models relatively quickly. It uses numerical maximization of the likelihood, but it's all coded in C++, so it's fairly fast. And then it has some nice functions for visualizing the model and for doing what's called state decoding. That's where you can take your model estimates and actually get the state sequence, which is often what people are most interested in. They want to know what the animal was doing and where. So it has functions for that. And it also can plot covariate effects and things like that with confidence intervals and all that good stuff. And lastly, it also provides diagnostics for goodness of fit. And those include things like pseudo residuals, which we'll talk about more. Yeah, so lots of good stuff in there. And I'll just go over these a little bit step by step. So first, to prepare the data in MoveHMM, there's a function called prep data, and it will accept either UTM coordinates or lat long. There are some issues if you use lat long though, because the way it calculates bearings and things like that. So, I mean, personally, I would recommend always using projected UTM, but you can use both. And what this does is just gets the data in a format for the other functions in the model. And then now it becomes an object that you can plot just using a generic plot function. In this case, we have an elk from a seminal Morales et al. Paper from 2004, where he and his co-authors first looked at these types of models. But it'll help you visualize your tracks by each individual, and then it'll give you summaries of the steps and the turns, which can sometimes be helpful for just understanding what's going on with your data. And it'll also give you some histograms as well. So for model fitting, the main thing for fitting models in MoveHMM, and the hardest part of all these, is that you have to specify starting values for the optimization. So there are some tricks for doing that. Sometimes trial and error is honestly the best thing you can do. You can also look at your data histograms and things like that. So sometimes you need to fiddle around a little bit with this, depending on the quality of your data. But once you've done that, you can then just use a single function, which is called fitHMM, and you just supply the data, the number of states you want, and then the distributions for your step lengths and your turn angles. In this case, we're using a Weibull distribution and a wrapped Cauchy for the angle turn angles. And that's it. And then it'll fit. And then when we print the results, you can get the summaries of your parameter estimates. We can get our intercepts for our state transitions, all that good stuff. Now, just a note. If I get the light here, yeah, okay. Okay, yeah, and just a side note here about these models. You can use something like AIC to choose the number of states, but what does tend to happen with these is the more states you add, the more favorable it'll appear to something like AIC. And there's a really great paper by Jennifer Pohl from a couple of years ago that makes the case that for these types of models, you really want to use biological intuition. I mean, don't throw out all your statistical, prowess and thinking, but they recommend using AIC only to compare like which covariates are important, not necessarily the number of states, and then also using pseudo residuals for assessing goodness of fit. Yeah, it is one downside of these models, though. If you keep adding states, usually the fit will keep improving, and AIC may just say there's 10 states here, and you could provide no biological justification for 10 states. So just a word of caution there on using AIC. So there are other choices for the distributions. They're somewhat limited in MoveHMM, but popular ones are gamma or Weibull for the step lengths, and that's because they're non-negative. And then for the angle distributions, like the von Mises is popular, and the wrapped Cauchy is also sometimes used. The von Mises is more analogous to like a circular normal distribution, so some folks tend to prefer that. Any questions? I feel like I'm blowing through this material here. Yeah.
What was zero mass?
What's that?
There was also the third parameter.
Oh yeah. So yeah, I think I said non-negative, and that's you have to include a zero mass parameter for the gamma. Any of these positive distributions, you use a zero mass parameter, which then accounts for step lengths of exactly zero.
Okay, so if the animal is sleeping, for example, that comes to that state, or
yeah, so that's a zero, and then so your zero mass parameter is trying to estimate like what proportion of all step lengths in the state are zeros. It's kind of like an occupancy modeling or something like that. We have your psi times your detection history, then you have 1 minus psi if it's not occupied. It's kind of like that kind of mixture. So it can either be zero, which has probability zero mass, or it can be whatever the step length is greater than zero times 1 minus the zero
I was
mass.
thinking like if the animal is moving or standing, so is it the same? Like standing is, then it's not two states, right? Because one state has no angle and no shape if it's just fixed.
Right. So yeah, but so within a state, let's say it's resting here. Okay. Let's say half the time when the animal's resting, it's actually not moving. Like that step length is zero. But then there may be some where it moved like a meter or two. Those will both still be resting. It's just that that distribution will be broken up into the steps of that were actually zero, and then those other ones that were just real tiny. Does that make sense? That's the question. Oh, and another thing that's nice about MoveHMM is you can specify a formula for your state transitions, and it's just your simple, like typical formula in R. So you could just use your little tilde thing, and then you can add your covariates in there, and then it'll make your state transitions a function of whatever covariates you put in there. So for data visual or results visualization, there is a generic plot function for FitHMM. And for these results, this is again one of those ELK. And we can see that it's probably very hard for you guys to see in the back. Sorry about it. But for the step lengths, there's one distribution, which we're calling state 2 here, which has much larger step lengths. And then there's another one, state 1, which has short. We would often call that foraging and transit, basically, because when we combine that with the turn angle, we see that the state 2, which is the transit state, has its concentration around 0, which means it's maintaining the same direction consistently. Whereas in the foraging state, which is state 1, the density goes down around 0, so it's more likely to change direction. Does that make sense there? Okay. So you can also, plot your tracks with the state assignments. So here we see we have our foraging states, just those little brown clumps. Yeah. There's what?
A little stick there.
Oh, wow. That is great. All right. Thank you. Here we like scratch this thing. Okay. So here we have our brown spots are our foraging locations. And then I was going to walk up here, but I was going to trip or pull something out anyway. And then we can see these kind of long distance transitory movements between foraging bouts in blue there. And this is what I was talking about earlier with this is something called the Viterbi algorithm, which is, Viterbi algorithm, where you can decode your states and it'll actually give you the state sequence for each track that you fit. So you can see here we have transit, foraging, transit, right? So you get that as output, and then you can also get the state probabilities. And these are different from the Viterbi algorithm because they actually give you the probability of being in each state through time at each time step. So for example, for the first observation, very high probability that this animal was in state 2, which was the transit state in this case. Yeah. So a very handy function is this pseudo residual plot function, plot PR. And this will look at your model fit. So here we can see, we're basically trying to see how normal looking are these pseudo residuals. They're very similar to residuals in just a standard regression. It's a little different here because this is a hidden Markov model, but that's the general idea. And here we see that, fit's not too bad. We hope that these residuals lie right along that line here. So we have a little lack of fit up here. And there's a little bit of maybe extra autocorrelation in our residuals here, but overall, that's not a terrible fit. So it's just a handy function for helping you assess how well your model fits the data. I haven't talked too much about covariates, so here's like a huge covariate example. This is also from the Morales et al. 2004 paper. But as I said, you can specify these simple R formulas for the state transitions. In this case, for the ELK, they threw like every conceivable distance covariate into their model. So like distance to water, distance to swamp, distance to whatever. And they fit all those in there. And you can just do that, just embed that within the FitHMM function. You don't have to, without having to custom code anything. So it's real handy for checking out different models with different covariates and things like that. And this is what we get. I'm not going to go through all these, but if it's negative, that means so we get to have two state transitions, right? We have a transition from 1 to 2, or a transition from 2 to 1. So if it's negative, so distance to water, that means the animal is less likely to switch from state 1 to state 2 as it gets farther away from water. Okay. We're not going to go through all of those, but so it just helps you identify which of these covariates are driving that behavior. And this is just a plot of some of those relationships. So this is distance to NTW, which I can't remember what NTW stands for, but we can see, you know, as a function of whatever this thing is, you know, our state transitions change as a function of that. Yeah. So that's really in move HMM, to getting at the biology is really through the formula function to say, you know, to see how are the behaviors affected by different covariates.
Quick, and maybe I missed that, but you provide the number of states, and then how does it make kind of a cluster analysis of the distance and turning angles, or how does it?
It's great. Yeah, it's a great question. You normally would have gone into that. So it's a likelihood function. So it uses the forward algorithm, which I don't know if anyone saw my talk on Tuesday. That was up there for like 2 seconds, and then Clark Rushing had I think had that up there as well. So it's a likelihood that is optimized. So it starts at those initial values, and then an optimizer tries to find the solution, but the maximum point of that likelihood.
The solution will be different if you have covariates.
Yes, the solution will be different if you have covariates. I mean, as long as if the covariates contribute anything, yes. In theory, if they contribute absolutely nothing, wouldn't change anything. Yeah.
And so you have covariates like switching from 1 to 2, but you can also stay from 1 to 1, right?
Yes.
And can you have covariates from that transition, or is it just negative of?
Yeah, well, like if you only have 2 states, it doesn't really matter, right? Because it's just 1 minus the, you know, the transition from 1 to stay in 1 is just 1 minus the transition from 1 to 2. But if you have more states, there would be situations where you might want to just model the probability of staying in the state, perhaps. And you can't do that in move HMM, but you can do that in momentum, which will be the next package we talk about. Yeah. And you can also use AIC. Again, don't want to use it to choose the number of states normally, but you can use it to see what kind of evidence you have for your state transitions. So you can use AIC here because this is, you know, it's likelihood based. So in this case, it's saying that the model with the covariates like absolutely blows the other model out of the water. So there's a lot of evidence here that the behavior is driven by those covariates here. Okay, so that was a real fast introduction to move HMM. Were there any other questions on that before we move into? Yes.
Yeah, I was wondering about the biological. Is there, or what would be the best way to interpret what these states represent? I mean, maybe I missed that, but like we define the number of states that we think there would be, but I mean, maybe it could even. I mean, it could be that those that what model determines as states are not exactly those that we had in mind, maybe.
Yeah, no, that's a great question, and yeah, that comes up all the time. And so, in that Poll et al. Paper that was up there earlier, and you guys get that from the slides. I mean, that goes into this as well. Usually, you have some as a like we're all biologists. You have some idea of what your animal is doing, and that's why folks often limit it to like maybe like 2 or 3 states that they know they can probably identify. Like, so foraging often looks like this, and then, you know, migration or transit looks like this. Those ones are pretty safe. But then as you start adding more and more states, the model will just start like it'll differentiate this type of foraging from this type of foraging. Yeah. So yeah, you have to kind of be careful and think about it. And then we'll get into it more once we get into the momentum, but you also can bring in other data types. So trajectory isn't always the best for inferring behavior, right? Like more than 2 or 3 states just based on horizontal trajectories, it can get a little dicey. So what you can do is bring in other data types like 3-dimensional stuff like diving or accelerometer data or foraging versus resting, all sorts of things. Yeah.
So this way you could also introduce maybe from some steps, you know what the behavior is, or you want to force the model to assign this to one state.
You can do that, yeah. Yeah, so in both move HMM and momentum, there is an argument called known states where you can just say, I know it is foraging at this time step if you want to do that. Great questions. All right, now we're moving into momentum. What's that? Oh, check chat. Yeah. Okay. Thanks, Devin. All right, so the motivation for momentum, I mean, MoveHM is a great package. But our data types are changing and we're getting, our biologging is getting more advanced. We have all sorts of things that these things are collecting now. So in our lab, for example, we now are getting location, but we're also getting like dive activity, we're getting accelerometer stuff, we're getting temperature stuff, we're getting all sorts of information from our tags. And MoveHM just can't accommodate those. We also are now dealing with more and more spatial temporal environmental data. So, you can go now and download, sea ice concentration for the last 20 years at a daily resolution. You can get habitat type layers, but all those things are very difficult to incorporate into these types of models. And so I wanted to be able to incorporate these things in order to learn more about animal behavior than just what the horizontal trajectory is telling us. And I also wanted a way to link movement and behavior to these spatial covariates. And when I say these are generalized multivariate hidden Markov models, that just means that if we're going to stay in the step length turn angle world, we're going to keep those components of the likelihood, but now we're going to start adding additional components that incorporate other data types. So whether it's accelerometer data or dive data, we're just going to be adding additional components to our likelihood so that we can make new inferences or better inferences about different behaviors at the time. So momentum is very similar to MoveHMM. It's got like many of the function names are the same, but underneath it's got a lot of stuff added on and extended. It's available on CRAN. And the vignette, I think now it's beyond 15 now. I think there's like 18 case studies in there, but there's a ton of examples in there. So if you're interested, I would encourage you to check out the vignette because we just don't have time today to go through too many of them. And there's also a paper from 2018, but again, that's also pretty short. So the vignette has got a lot more stuff in there that is really aimed at practitioners who want to use this for their data. So yeah, if I had one statement to make between the two packages, MoveHM is easier to use, but Momentum is way more flexible. So if you have a very simple problem and you just have like two states and maybe a couple state transition covariates, MoveHM is just fine. But if you have spatial covariates or other data streams, Momentum would be more accommodating to including all those different data types. Yes.
Sorry again. If you have irregular sampling or like tag doesn't work in the night and works only in the day, so can we do this?
Yes. So yeah, I guess discrete time, like maybe we do this so often that we forget to say that discrete time model in this case means the observations are regular in time, so equal time interval between observations, and that often is fine for like terrestrial data sets, like those are often very regular. But for marine systems, oftentimes you don't get an observation every hour or every 15 minutes. You get it when the animal is at the surface long enough to get a fix. And yes, we could do things to account for both missing data and irregular data and then measurement error as well. And we'll get to that. That was another thing that Momentum was developed specifically for. Yeah. Good question. So the additional capabilities of Momentum relative to MoveHM are that it can not only do correlated random walks, it could also do biased random walks, so movement towards targets or areas of attraction. I'll talk more on that in a minute. You can have as many data streams as you like. So if you have all sorts of auxiliary data streams, you can include them all in a single model to help inform and distinguish those different behaviors. It also has a lot larger choice of distributions for your data. So we've got pretty much every one we can think of in there, but we also could add any if you guys have any requests. It's pretty easy to do that. A big difference is you can have covariates on all of the parameters. So not just your state transitions, you can have covariates on, say, your step length distribution parameters. So the distance you travel could be related to, say, temperature, like if you don't like to travel too fast when it's hot, things like that can be incorporated into Momentum. You can also constrain your parameters however you like. And then you can incorporate covariate raster data pretty seamlessly. So you can you can take the location at any time, pluck out your spatial covariate, and then use that either as a covariate in your state transitions or also on your movement properties as well. Yeah, it allows for centers of attraction, potential functions, which is what I talked about on Tuesday, if anybody was there. You can incorporate those, and then you can also do group dynamic movement models. That's where animals kind of move together. You can do those in momentum. And then you can get kind of funky and do like cosine models, which are cyclic, or you can do even spline models in there. So very, very flexible formulations. And related to that question we just got, it can do multiple imputation, which is a way of dealing with irregular sampling and with measurement error. And it can do all that in parallel, which so for when you're doing this step, the parallel processing is really handy because everything in that process is parallelizable. What is that thing? Embarrassingly parallelizable? Yeah, something like that. Yeah, embarrassingly parallel. Yeah. So I alluded to what a biased random walk was a minute ago. Here we just have our simple random walk again on the left. And this is an example of a biased random walk where we have basically two centers of attraction and the animal will switch between being attracted to one or the other. And then we have another state in this case where it's just kind of exploring. But this part here where it's moving that way or is attracted that way, that is a biased random walk. That's also called drift. And that could be due to like gradients. So like ocean currents could be pushing it. You can incorporate that into the model. There could be wind. You can incorporate that into the model as well. Or it could just be like a known haul out or rookery site or a nesting site, things like that. So it allows for a lot more interesting like biological hypotheses to be addressed once you start putting it into the models. Also include repulsion too. I've yet to see this done, but you could have like repulsion to predators and stuff like that, which would be kind of neat, but yeah, it can work either way. And here's a little pitch for incorporating additional data beyond just the horizontal trajectory when you're doing these types of models. And that's because it can be hard to discern more than two biologically like meaningful states based on trajectory alone. So if you incorporate additional data types, it can make it a lot easier to differentiate them. So a great example and a kind of a classic example is one where we have what we have here, where if you were to analyze this as a three-state model with just based on the trajectory, like it wouldn't be able to differentiate the resting state from the foraging state because they look virtually identical based on the horizontal trajectory. But here, by including just dive data, is it diving or not? You then can differentiate the resting when it's not diving versus the foraging when it is diving, even though those horizontal trajectories look almost identical. So it can be really handy for distinguishing those different types of states. And here's the workflow of the package. We start with some data. And then you can either have measurement error or not. So if you don't have measurement error, you can then just go straight to the prep data function, which gets your data ready for analysis. And that produces what's momentum data object. And from there, you can then plot, or then you can fit a model using fit-HMM. And then from there, you now have a momentum object, and then you can do further post-analysis like plotting, AIC, or pseudo-residual plots, things like that. It's a little different if you have measurement error or your data are irregular. Then you have to follow this path, which is much more complicated, to crawl wrap, which is a wrapper function for crawl, which is a continuous time correlated random walk. And Devin will talk about that in much more detail. But what this does is it provides a way to simulate realizations of the path that accounts for your irregularity and your measurement error. So you can think about it, it's integrating over all the possible paths that the animal could have followed if you had been able to observe it. But what it does is it does that and produces it then in a regular discrete path that you can then fit repeatedly using just the fit-HMM function. Okay. And then after you say you've done this 30 times, you've drawn 30 realizations of the track, you fit the same model to all 30 of those different tracks. You then can pool them together for joint inference across all of those imputations. Okay, so it's kind of like a trick to not have to go into like the the Bayesian world or something like that, or a hierarchical model that accounts for the measurement error in a more like exact way. So this is an approximate way to handle that measurement error and temporal irregularity. There's a few papers out there that have looked at this specifically, and it generally works pretty well. It's just you can't have like extreme error relative to the movements of interests.
Is that where the parallelization comes in?
Yeah, so this is embarrassingly parallelizable. Yeah, because each one is independent. Once you've simulated your however many tracks you want, you can then run them all in parallel. Yeah, so it's a lot faster. Yeah. Okay, now for some examples. Let's ask some questions. Yeah, sorry if this is going fast. Short afternoon, yes.
If you use different kind of data like accelerometer data and GPS, they have to be synchronized. For example, if you have GPS every half an hour, but you have acceleration every 5 minutes, so how do you do that? Do you like do an average of acceleration and put them to each GPS location?
You could do that. Yeah, I don't have like the correct answer for that. Yeah, you do have to try and line those up however you think is most reasonable. So sometimes we have that problem with dive data and location data. So sometimes if we have like bin dive data, we will assume it was, the same for like an hour, like let's say observations every hour or something like that. Yeah.
In your number 2 example, did you have the dives in that?
Yes, dives are.
Okay, so the number 2 example, in that data set, we had, however many dives the animal does, it just dives. So when you line it up, you did it hourly or something. One metric you could use is say maybe number of dives within that hour. So each hour then lined up has a covariate data. Yeah, so you have to summarize that other data.
Yeah, and like so with the accelerometer data, you don't want to lose too much resolution there, but you may be able to use some summary over whatever the interval is for your locations, say, and use that to kind of summarize what the accelerometer data are telling you from one location to the next. Or the other option would, depends how high resolution your accelerometer data are. You could impute the locations more closely, so you can use all of that accelerometer data. Yeah. Okay, so just kind of run through a few quick examples here. This is elephant data from this paper here. It's in Africa, and here's what the track looks like. Came from Mevin, but I don't think they've done this type of analysis with it before. So this was just kind of made for a nice example in the paper we wrote for here. This is an example of what the data look like. We have this is common to all data for momentum. You're going to need to have an ID column that identifies your individual. You have your coordinates. They can be called whatever you want, but we're calling them X and Y here. And then we have 2 covariates. We have temperature and time of day. Okay. When we run this through the prep data function, we're telling momentum that these are lat long coordinates and that our covariate names are temp and time of day. And then what this will do is calculate step length, turning angle, and then it knows not to mess at all with your covariate names. That's why we're identifying them there. And then once we've prepped our data, we can then use these generic plot functions to plot our track. We can see histograms of our step lengths and our turn angles, and also to see those through time. We can see that we have a lot of turn angles concentrated around 0. And you can't really tell here, but it looks like there might be kind of 2 distributions kind of mixed together on the step lengths, but we'll see about that once we fit some models here. So we're going to specify a gamma distribution for our step lengths and an angle or a von Mises distribution for our turn angles. And then, as I said before, the hardest part really is you got to come up with starting values here. In this example, it's not too hard because the data are pretty good. But if your data are kind of sparse or not, if it's not real clear what the 2 states are, you may struggle a bit with that. So that's probably the hardest part. And then for the initial model, we're going to say we have two states with the distributions we've just defined, and those are our initial values. And then the package will spit this back at you so you can make sure you don't, this is what you wanted before you let it run. And then here what the results look like. It's very similar to Move HMM. One thing that I'll note here is we fix the mean of the angle distribution to 0, and that's what gets us at a correlated random walk. By fixing that to 0, you can then get a measure of how strong the directional persistence is, because you want it relative to 0, because that's when it's traveling in a straight direction, right? So the higher value here suggests that state 2 is the more directionally persistent. Okay, and these are what those distributions look like. We can see we have some directional persistence in both states, but this state 2 appears to be the state with more directional persistence. And if you can see it, it has longer step lengths as well, right? So that might be our transit state, and state 1 is our foraging state. This is what the plot looks like, and that's consistent with what I just said, that yeah, we travel faster and farther and straighter when it's in that second state. Here's just another plot using the satellite imagery. And here's what our pseudo residual plot looks like. So, you know, you might look at this and think, okay, it looks okay, not too bad. You know, maybe we got some lack of fit in the tails there. But then we got this little thing right here. Does anybody have any idea what might be going on there? I'll give you a hint. The 24 is about right here. So yeah, if we look at that, we zoom out there, we have what appears to be a 24-hour, you know, periodicity in there, which is really bad. It's, yeah, huge. So what can we do in that situation? One option would be to use a periodic function on our state transitions, okay? So we're gonna now say, we're gonna throw temperature in there too, because the temperatures kind of fluctuate each day. And then we're going to use what's called a cosigner model with a 24-hour period. So that just creates a periodic relationship through time. And if it's on a 24-hour period, that means that it has to reconnect at the edges there as it goes through, right? So it's just creating a cycle of sorts. So we're going to put that on our state transitions and then fit that model. Oh, and sorry, we're also going to add the same model to our step length. So now not only are state transitions a function of temperature and time of day, but our step lengths are also going to be a function of temperature and time of day. Okay. So if we compare the AIC for those three models, again, we have a case where the model with the temperature and time of day on everything just blows away the other models. So it clearly explains a lot more of the variation. Play is here now.
Not sure. I think it's between there.
Okay. So here's what that looks like. So these are plots. Since we have covariates on our step lengths, this is just for fixed values of those. But we see the same pattern where state 2 is the faster, more directed state. And maybe more interesting, there we go. And this is what the state transitions look like as a function of temperature. So state 1, if you remember, was our kind of like foraging state. Is that right? Foraging state. State 1 is foraging, yes. So so as temperature increases, it's more likely to stay in that kind of slow moving foraging state. And then it's more likely to switch to state 1 as it gets hotter, right? And that can make intuitive sense, because if it's real hot, it probably doesn't want to travel fast in the middle of day in this case. So it's reflecting that. And similarly with this plot here, it's a little more obvious here that the high temps, it tends to want to move less. And with that model, it's greatly improved that residual autocorrelation. It's still not perfect. You can see there's still some there, but it's definitely doing a better job. Yeah. Any questions on that example? We just have one more and then we can take a break. Okay. So now we'll move into the northern first field example. And this is going to be one of these multiple imputation examples. And depending how we're doing today, like if we have time, I do have another one of these if we want to get into this. I also have a bigger example of the talk I gave on Tuesday with how to do that in the package if you guys are interested. So we can maybe see how everybody's feeling like that. It's been a long week, but if we're feeling it, we can get into more detail on that stuff. So anyway, this example is with a northern fur seal. We have two data types here. We have not only our locations, but we also have the number of foraging dives per hour. But these location data are irregular in time. So what we need to do in order to analyze these together in a discrete time model is get our locations to align in time with our dive data, which is hourly. So we need some way to regularize this track into 1 hour segments so that we can match it to our dive data. Okay. So this multiple imputation approach is a way to deal with either irregular sampling and or measurement error. Sometimes with these marine species, the error can be a lot worse than it is on terrestrial systems. And also because they dive a lot, you just miss the locations that you want. So it's generally a bigger problem in marine systems, but it is still a problem that needs attention even in terrestrial systems. But the general idea is that we're going to use a continuous time model that Devin's going to get into to simulate a bunch of tracks that could have been produced given the observations that we had. So the idea is like here we have a big gap in our observations. It's essentially going to integrate over every possible route it could have taken during that time step. Okay. And then we're going to get predictions for each of these simulated tracks. We're going to get hourly predictions of its location. And then we're going to fit the HMM to each one of those tracks. And that's the multiple implication part. Okay. And the way we do that is through the crawl wrap function. It is just a wrapper for the crawl package. So it just does the step where it fits the model. And again, that model accounts for measurement error. So that's the main thing crawl is doing for us there. And then it also allows us to simulate those realizations from the track. So the crawl wrap function just does all of that in one wrapper function. And then it gives you this as your output where you can look at your predicted and observed locations and make sure everything looks okay there. Now that we have our data the way in the format that we need it to be, we can specify the model that we'd like to fit. So in this case, we're going to use a gamma distribution for the step length, a Rapkowski for the turn angle, and a Poisson for the number of dives. Okay, so this is a multivariate hidden Markov model where we're using our location data to inform steps and turns. And then we're going to add in our dive data to hopefully help us get at foraging. Okay, because these are the number of foraging dives. If we didn't do that step without including the number of dives, there would be just a mix of foraging and resting, because these animals tend to rest at sea, and you would have no way of distinguishing those. Okay, so as before, we just specify the distributions that we want. And in momentum, everything is just a list. So everything's like list arguments. So all the different arguments that you can specify in the main functions are just lists where you identify what you want for each data stream. So in this case, we have 3 data streams. Then we get our initial values. Don't ask me how I got those. And then we specify just our list for those initial parameters. And then in a single function call, we just use it's called MI fit HMM. That's for multiple imputation. Just in that little bit of code there will then fit all 30 realizations in this case, fit the same model to all 30 realizations. And all we need to do is say the number of states, the distribution, and the number of parameters, and then it'll do the rest for us. And then as it runs through, what it'll do is it'll run all these in parallel, and then it'll do the state decoding for each imputation. It'll do the state probabilities for each imputation. And then it'll pool all of the results into a single final estimate and calculate the variances and covariances pooled over all of those things. And I think it also will get you a 95% error ellipse over your location. So you can get an idea of how you're doing relative to the measurement error of your model. So it'll show like how much error you have. And these are the model fits in this case, the pooled the pooled inference across all the imputations, where the step lengths and turn angles are kind of what we would think they would be for transit versus resting and foraging. The main thing here is the main thing differentiating them though is the number of dives, where foraging state has a large number of dives, the resting state has absolutely no dives. And then it looks like there might be some sneaky little diving during the transit state as well. And that is consistent with these guys. They are known to sometimes wiggle down and get a snack while they're going at high speed. So that wasn't too surprising. And then you can see here just the state sequence plot. And then you probably can't see the air ellipses, but it shows you, wherever there was a large gap in our observations, we have these kind of larger ellipses. And we're accounting for that uncertainty via the multiple imputation. We'll do them both. Yeah.
It looks quite straight. So would you come to different conclusions if you would just make a straight line and be much faster in computing the?
Well, you do it in parallel, so it's not faster. But yeah, no, it takes the amount of time that you have. If you're doing 100 of them, maybe be faster. But yeah, no, that's a good point. If you did draw a straight line, in this case, if it's a perfectly straight line, it wouldn't make much difference. But if depending on the time, it's hard to see how much time is going on there. Like if there's a larger time gap, the animal in theory could have gone out here and back here, or done like some foraging and come back and you just missed it. If you just assume it's a straight line there, it's going to miss that potential. So by doing by simulating from it, you're accounting for basically all possible paths you've gone through, right? So it really depends on the length of time. So like I wouldn't be comfortable with a straight line through this stretch. It looks like it's a small stretch, but it's actually a pretty big stretch. And then the realizations do kind of take it out kind of farther. So, in this example, maybe it would make a huge deal, but especially if you're like relating those positions to spatial covariance or things like that, I would want to make sure I'm covering all the variability in there. Yeah, good question. So with you, Doctor, we have one here in the word.
And did I understand correctly that also like uncertainty in the locations, in the individual locations is also taken into account?
Yes.
Oh,
yeah.
And how does that work, or how? Oh, yeah. Okay.
You can wait for the
Yeah, I think that's that. We'll just wait. We'll hold off on that. Yeah.
But you also classify the behaviors in the unobserved part, right?
Say it again, sorry.
So you also classify like whether it's walking, whether it's transient or feeding in unobserved part.
Yes.
And that would kind of by default that would impute most present state.
Not necessarily. So there's a fair bit of persistence in the states. So for example, the gaps here, once the animal is transiting here, the state transition probability out of transit is very low. Okay, so if it's in this state and then it hits a missing stretch, it's probably going to keep it in the transit state, even though the transit is not the most frequent state, right? So yeah, I mean, it's not going to magically pull out the whole sequence, but it'll go with what's most likely. And that's the whole point of this, is that you're going with what is most likely, given I've gone through every possible path that it could have been in, what is the state sequence through that? Yeah.
It can also estimate that it was moving and then it transited to the foraging state during unobserved part, and then it comes out of unobserved in the foraging state, for example, right?
I'm not sure. It's unlikely to probably it's gonna, that gap is going to be. It depends how much measurement error and how long the missing period is. I mean, some of those tracks, in this case, this wouldn't happen because the gaps aren't that big. But you can have some of these imputed tracks that go all the way out here and come back. Like crazy stuff like that, and then it will be traveling unless you have other information in there. It's going to think it's going about the same speed through that. And if that speed lines up with one of the states, it is likely to assign it to that state. But yeah, this isn't exact. I mean, this is like a way of approximating that. If you want to really do it exactly, you would need to do a single stage model.
That was Brent's dissertation, so he didn't read his dissertation.
with that accelerometer data, do you then just provide X, Y, and Z as covariates, or do you?
What was the first part there?
If you have an accelerometer
Yes.
on the animal,
Yeah,
you have X, Y, and Z.
you could model those directly if you want, yeah.
You would put them in as covariates, or do you pre-process them so how to?
So if you have X, Y, and Z, I mean, you could use a multivariate normal and have it be like a random walk type thing if you wanted. I mean, otherwise people have all, like, you can actually model like 3D accelerometer data as a multivariate normal with a 3-dimension multivariate normal. And then you could have correlation in the X and Y or Y and Z if you wanted. So you could do that. Other people summarize their accelerometer data into some single metric. Yeah. There's lots of different ways you could do that. I'm really interested in the multivariate normal. I haven't seen anybody do that yet, but I think it'd be kind of neat.
I was just wondering the step lengths, turning angles, and then the diets. Those are all conditionally independent given the states. Does one often need to worry about that or Hooten dependence there? Or?
Yeah, I mean, folks have looked at that. Usually it's not a huge worry because it's conditional on the state.
Yeah,
you're right. But there are some, like someone has cooked up a scenario where that was a problem and they wanted to have a non-independent. And one way to get around that is to model the positions. So you could have, instead of doing step length and turn angle, you could just model X and Y as a multivariate normal with a correlation term. Right, and then then they can be correlated. It's just some folks find that harder. Like people like step length and turn angle because they're easy to interpret, right? But you can do the same thing with a normal random walk on the locations themselves. Great questions. I also feel like my PhD defense.
Yeah.
We're gonna say something?
No.
Okay, how are we doing on time? I got a few other little short examples, but we can take a break. It's already been almost 2 hours. Do you guys want to take a break?
Yeah.
Okay, let's do that. I think there's some refreshments in the back, yeah? That's okay. So I think we have our schedule. We were planning to go till 5:30. Is that sounding like horrific for everybody, or is that, do we want to go to 5:30? Then I could talk for a little bit more, and then Devin could take the floor like 4, but then we would go to about 5:30. So what do you guys think?
I feel like you guys should do what you prepared. You put a lot of effort into this. It's really great. But that's my 2 cents.
Okay, then yeah, as long as there's no strong objection, we'll do that. And then we can also, I think we mentioned in the email, we can take these discussions to town if we want or wherever, because we're here till tomorrow. So happy to continue that on afterwards. So with the 20 minutes or so that I have remaining, I could either kind of have like 2 choices with 20 minutes. I have another like bigger multiple imputation type example that involves like a whole bunch of data streams, like variable measurement error, regular locations, things like that. It's a bearded seal example, and it involves spatial covariance. Or I also have slides prepared that show how to do the Longitudinal diffusion model in momentum, and that gets into more advanced model specification in the package.
Hey, Brett, before you go on, this is Mevin. It sounds like we lost the sound online and maybe the video with Killian's computer. Okay, I can kind of hear people again.
I know what to do. I need to talk about it. We're gonna go all the way to.
Yeah, I know, but on the straight, the left, don't left. Nothing to them.
Okay, can everybody online still hear us?
Yep, and we can see you again too.
Can everybody online still hear us?
It's watching.
Yep.
I can see Hooten, Worthington. Do you guys hear us? Okay, Mevin can hear us. Okay, I think we're good. Okay, so this is just going to be like an applied example with momentum, and this is using some bearded seal data that my lab collected. These guys are really, really hard for us to catch, and we only got like 7 after years of effort and stuff like that. So these were very important to us. So we wanted to extract the most we possibly could out of them. But they imposed some difficulties. So these are kind of your classic complications with telemetry data. We had big measurement error because we're up in the polar region. Lots of missing data because the animals, dive for a long time, and then so we had to track stretches of just no data. And when we did get locations, they were irregular in time because we only get fixes when the animal is up out of the water long enough. And bearded seals are ice-associated seals, so they tend to haul out when they have ice. But then often in the summer, they will not haul out at all. Those you can see the whole time. And then you get locations then if you're lucky enough for them to keep their head out of the water for a little while. So historically, I mean, usually these weren't even kind of the realm of hidden Markov models. And the reason for that is because with these complications, you often had to do what were difficult and very computationally demanding methods like MCMC to fit models like this, or use something like an EM algorithm. So that was a big part of the motivation for the multiple imputation approach, so you wouldn't have to go down these routes. Okay, so for example, I used to do all the MCMC stuff, still do sometimes, but this one seal took 5 days to fit using MCMC. That was coded in C. And then this bearded seal took 3 weeks for 7 bearded seals. So yeah, it took a long time. Now I'll admit I'm not the best MCMC coder, but that's still a very long time. So the multiple imputation approach is one solution to try to kind of avoid having to go down that route. And several papers have now demonstrated that it works pretty well as long as your data aren't like really sparse when it comes to the things that you're interested in. So if you got these huge gaps in time and your measurement area has, if your study's in Alaska, but you have locations that are in China, it won't save you from that. Nothing probably will, but within reason, this will do a decent job if your data are at least fairly decent. It's just you can't do it with really lousy, what I'll call lousy data, but that's not really fair to call them lousy given how hard everybody works to get their data. So, yeah, it's just kind of what we have, the world we live in. But anyway, we already saw this slide. We're going to fit a continuous time movement model that Devin's going to give all the detail on, and then we're just drawing all these realizations, and we're going to fit the model to each one. The way that the multiple imputation works is it just uses kind of classic statistical theory, where if you take up all the estimates and the variances from all the individual fits, there are a well-known formula to get your overall estimate and your overall variances. So then that's what momentum does when you fit the multiple imputations. It'll then calculate the overall maximum so then you get things like confidence intervals and your error bars, all that stuff. And I'll do the same for the state assignments. So it'll do the Viterbi algorithm for the state sequence for each imputation, and it'll pool those, and it'll pool the state probabilities. So you get all the things that you normally get from a single fit, but it's just pooled across all the fits. And the nice thing is that it's, again, embarrassingly parallelizable. So if you have enough cores, you can do, as many imputations as you want in as much time as it would take to fit one model. So here's our bearded seal example. I think, finally we got a picture of an animal here. So you can see the tag there on its head. This is what the tracks look like. So we have 7 individuals, and this is Alaska. They're all tagged near Kotzebue, and there's a lot of locations. So we got over 50,000 locations for them. And we also have both dive data and wet dry, like conductivity data, but it's in 6-hour bins, which is kind of long. But the saving grace, I guess, is that we're talking about animals that travel a great distance. So we think that the 6-hour bins are reasonable for inferences at a larger spatial scale. But yeah, if you want to get a high resolution behavior, a 6-hour bin is not a good thing to work with. We had plenty of missing data. About 18% of our time steps, 6-hour time steps, didn't have locations. And we had measurement error. We had both Argos and FastLoc. Now the FastLoc is pretty good, but it still can produce errors larger than you would think. And then Argos, can be all over the place. If you guys have ever worked with Argos data. So the idea was we were going to try and combine all of our sources of information. So when we combine all of our dive data, our wet dry data, and we're also going to use things like sea ice and things like that to try and fill in all these missing gaps to get an overall picture, and then identify 6 behavior states from 7 data streams. So this is a big example here. And the states we were interested in were whether the animal was hauled out on ice, whether it was resting at sea, whether it was hauled out on land, which is pretty rare for this species, but they do sometimes do it. And then we wanted to identify 2 types of foraging, one at midwater, and we could get at that from the dive data, and then benthic foraging and transit. And benthic foraging really was the one we were most interested in, because this is a benthic feeder that primarily spends its time at the bottom digging its fate in the dirt, and that's why it has these kind of characteristic red faces. It's because they're digging down there for bivalves and things like that. So we have all those data streams. So we have our kind of our typical gamma and Rapkowski for the steps and turns. And then our dive data were like the proportion of each 6-hour interval spent diving. So we used the beta distribution for that because that's bound between 0 and 1 because it's a proportion. And then dry time was the amount of time it was dry. So that's also a beta. And then we also use sea ice cover as a data stream. So each cell would have a certain percentage of ice, and we modeled that as a beta distribution as well. That's kind of as a proxy for the hauling out on ice. So that's what that's trying to get at. We also had land cover, which is kind of doing the same thing for land instead of ice. And then the number of benthic dives. So this is the one that's really helping us identify the benthic origins. Yeah, and this is just what some of those things look like for one of our individuals. So yeah, I told you guys this one was complicated. So here are the relationships that we would expect from our 7 data streams. So, and the ones that are highlighted in red here are kind of the ones that we're like anchoring on for each state. Some states are only informed by 1 data stream, but then other states are informed by more than one. And again, our most important one is benthic foraging. So we would expect both the dive time and the number of benthic dives to be higher when they're benthic foraging. So we're going to key on that in order to identify where they're doing their benthic foraging. Okay. Any questions so far? Yep.
And so your mask is spatial mask is X, Y, and time, right? Because the ice cover changes over time. Or
yeah, so that's a time varying covariant. Yeah.
Oh.
Yeah. We have an animation at the end. That'll be I'll show it. Yeah. So the basic steps again, this is our raw data. So you can see it's kind of like spaghetti, and you can see the measurement error that we're dealing with, right? But once we run it through crawl, and Devin will give us the details, this is what we end up with. So the tracks seem much more reasonable, and we've dealt with that measurement error. And we've also imputed at 6-hour intervals. So we now have data that are ready to be used in a discrete time in markup model. So when we do this and we use the MI-fit-HMM function, I won't get into all the details here, but we did some things with constraining parameters that we don't have time to get into, but that's done through this DM argument, which is short for design matrix. So momentum allows you, if you guys, I guess familiar with program Mark, I figured it's Euring, you guys know Mark. So it's just like the design matrix to Mark. So you can constrain parameters by having a common intercept, or you can have an individual covariate, things like that. So you do that with the DM argument. And then what we get out of that is then our pooled state estimates for each track after going over all those imputations. Okay. And there's some interesting things we see. The blue here is where they are doing the benthic foraging. And then the red is where there's some ice haul outs. So you can see like in the usually up here in the summer, and there was some ice way up there when I went out there. So you know, just looking at it like that, it looks like it does a reasonable job of identifying you know, the ice haul out state. And then based on the number of dives, benthic dives, you know, we didn't get a lot of benthic dives in any other states. So we feel like it did a pretty decent job of identifying the benthic foraging. This is a figure from a paper comparing what you get when you do multiple imputation, which here is MLCB. I don't know if you guys can read this in the back. Or sorry, MLCB is the full, like the 3-week Bayesian version. Singles, when you do a single imputation based on the best track, and this relates to your question about what if you just, if it's a straight line, that's kind of what that approach is. And then MI is the multiple imputation. And they're pretty similar. There's some little differences here and there, but the multiple imputation is more consistent with the full single stage Bayesian approach than is the single approach. So in this case, it seemed to do a pretty good job compared to the, so the 1 hour model did a pretty good job compared to the 3-week model. So, yeah. Okay, and I think this is our animation. If I don't know if we have the permissions. Did you do the permissions?
Yeah, but we're in preview, not Adobe.
Oh, can we switch to Adobe maybe?
Yeah.
Oh, thanks. This is the grand finale. It sounds
So why didn't you use spatiometry or something if that's like foraging, like correct your?
It does. I didn't get into that. Yeah.
Okay,
Yeah.
Which one is it?
That's the discrete multiple imputation.
Oh, okay.
Yeah, while we're doing that, so we have the location, and then we have like the dive, and then we know what the depth is, and if the depth was at, you know, the bathymetry level, then that was considered a benthic dive.
Yeah, okay, cool.
Any other questions? We have 3 minutes before Devin's due. Is it slow?
Yeah, it's just.
So those data are messy, your raw data.
What was that?
The raw data are messier than I would have expected.
Yeah. So here we go through time. This is the sea ice here, where I think I reversed the colors here. Where I had it anyway. The coolest part of this, I think, is coming up in a second. Oh, right there. See that? So it turned red. It turned red when it hit the sea ice. But anyway. Yeah. So that's pretty much the whole point of this animation. Anyway, yeah. So this shows that, you know, that some of these covariates are time varying, which isn't a problem because the package will do that for you. It'll take the date of the observation and extract that from your rasters. So as long as you can build the raster stack, it'll pluck the value for you. Yeah.
These are all 7 individuals combined.
Yes, and they didn't always overlap in years. So sometimes there's only like 1 or 2 or 3. Yeah. Yeah, and then they come down in the winter. They'll start coming down here in the winter.
What's your cell size?
These I think they're not that. I mean, this was done a long time ago, so I think these might even be 25 by 25. Yep, there they go. The ice is coming in. It's winter.
They swim under the ice.
Anyway, you get the idea. And then we took these, then we identified the benthic foraging in a specific region where we had collected a lot of trawl survey data, like different prey species. And we ended up being able to identify kind of this like hotspot along the coast and a little point over there. And this is just using like a resource selection function, like what Mevin was talking about earlier. But yeah, it appears like in the summer, there's kind of like a smorgasbord things to eat right around there. So so yeah, we're using that you know, to help inform policy and things like that in this analysis. But anyway, we wouldn't have been able to do this unless we've been able to identify when the animals were foraging. So I think that's probably good. I think it's time to get Devin up here. So yeah, it's 4 o'clock. So this is just a little example with a loggerhead sea turtle, showing that you can, you know, model movements as a function of like sea surface current. So that's what's happening here, where this animal kind of just catches onto this one and then cruises out and then does foraging. So those are all things that are hopefully made easier to do through the package, because you can just give it your vector of ocean currents and then it'll do the rest for you. Yeah, so you can do that. What would have been a fairly complicated analysis on your own in just like a few lines of code once you've got your covariates together. There's another plug for our book, and then yeah, that's it. Yes, that's it. So unless there's more questions, we can move on to continuous time.
With a question online.
Yeah, well, answer question if you like.
Well, he's bringing that up. I was gonna ask about sample size. So the graded steels, I was thinking about multiple imputation and roughly how much missing data you can have relative to good data.
Yeah, so that was why we brought in the 6 other data streams. So there was a lot of missing data, like it was about 20% of missing location, but they weren't like these huge stretches. They were like here and there, mixed in, yeah, but fairly regularly. And then so the idea was try and fill those in generally with the other data streams, and the hope being that then you won't miss the big transit movements if you have a missing location here or there because they're moving at such a fast speed. And then the other covariate or other data streams will then fill in like it's, you know, benthic foraging, you know, when you don't have a location and you come back.
Yeah, and then you impute the rest.
Yeah, you do the locations, yeah.
Okay, so in the next up, did we want to do another break at some point, or we could do it after I do this one?
Yeah, maybe like
We can accelerate the breaks as we get closer.
yeah, maybe like 45 minutes
After I do this.
Yeah, we can see how everybody's doing.
So I may have probably a few other topics, so there's potentially more here than we want to get through. So I may give you guys some choices, and I do have some bonus material at the very end. It's totally unlisted anywhere if you guys want to talk about it. As I was watching all the talks today, I thought that one thing that we had mentioned, and Mevin actually mentioned it briefly, is now that all these models we're fitting are to individual animals at a time, right? And so if you wanted to know, if you want a population inference for that, what do you have to do after you fit, you know, these nice complicated models to 20 animals? What can you say about bearded seals, or what can you say about? So if we have time, or if you guys tell me, yes, we really like that, and I ditch another topic that we're going here, we can go to that. It's like a real short, I don't know, maybe 15-minute thing on some stuff that Mevin and I had been working on for population inference, sort of a quick version of it, of the more elaborate one he mentioned in his slide and the thing. So as we get further on, or after the next break, we can figure out what we want to do for that. Okay, so start now with continuous time models, and Mevin had mentioned, I want to make sure I'm not standing in front of the camera, that a lot of these continuous time models are basically stochastic differential equations in some way. So that's how we get around. Essentially what you're modeling is the rate at which I changed my location. So that's really what we're talking about modeling. And for this, I'll be focused on using the R package crawl that I wrote years and years ago. So it's not nearly as fancy as Momentum. Because those Brett and Teo work on that all the time, and this was kind of got relegated a little bit, but it basically models that continuous time correlated random walk. So it's persistence in there, and we can do different things with that. Let's get going. The first thing I want to mention is that main collaborator I have in sort of keeping this package up to date and things like that is Josh London. He's a biologist that works in Brett's program, collecting those bearded seals. So, and he's great at keeping me up to date with tidyverse and all this, the GIS stuff in R and stuff like that. And he is great at breaking stuff that I thought was rock solid. And so I can just give it to him to analyze some data, and he'll come back and say he broke it. You have to fix it. So just give him some acknowledgement on all this. Okay, so let's go over a little bit of background before we actually dive into what it's doing. So we know what actually the stuff that crawl is doing actually isn't overall that difficult of a thing to grasp, but maybe converting the stochastic differential equation to something that's actually useful to conceptualize is something that we can do instead. So let's talk about, and this is something that Mevin had already mentioned, some of this notation. I'll refresh your brain, because actually this time we will be thinking about location error and how we can model that as part of this. So we have our S's are the actual observed locations. So for that bearded seal example, they were those locations that are all over the place. Tau are our times that we observe them, 1 to n. And the mu's, as Devin mentioned, could be the real true locations that we probably don't ever see because we have those noisy observations. So there are some true locations out there at those times. The thing with correlated random walks is you're basically modeling the velocity. Devin had a slide where he showed that u at time t is kind of the integral of that velocity process. So think about there's a continuous process of velocity that the animal's moving, and it's really just represented by very short step segments of time taking little steps, right? And for me to get to the other side of the room, it's the sum of all those little steps. And so there's a velocity process that we're going to integrate up, and it's just summing all those little steps as we go. And then we can sort of mash all these things together into this alpha here. That's just collecting everything together, and we'll call that the state of the animal. So the state is their location and velocity at a time. So if you can think about that like the hidden Markov model, the behavior state. So there's a state here just as well. There. So I'm not going to give you the differential equation form because it's, if you're coming up with a statistical model, it's not very intuitive because you're talking about derivatives of Brownian motion and stuff like that. And if I just want to fit a model, sometimes it's not as intuitive to think about, okay, what are the pieces that will go in, where do my data go, and things like that. So we're going to talk about it in terms of this conditional formulation. It's given the state at the current time, so it's like a Markov process. Given the state at the current time, what will be my state at some time in the future? Right, and it's continuous time, so delta here doesn't need to be regular. I can pick whatever time into the future I want. 1 second, a day, an hour. We're just talking about where will I be in the future? And so we can represent it, that stochastic differential equation, by these terms here. So for this, the velocity where I'll be, or the speed and direction I'll be going, is just the velocity I was before times this sort of term here, plus a random component, right? That's just going to depend on the amount of time I'm talking about. So all it is, it looks a lot like an auto-regressive AR1 type thing, right? Where I was before times a little fudge factor in this plus independent random error. And along with that, the location will all be where I was before, right? It's like an autoregressive. But it's got, and we have the same, we have an error term, so it's kind of like that random walk or autoregressive. But the thing we have additionally in here is this drift term. So it's going to depend on where I'm going, right? If I'm running in that direction a little bit into the future, chances are I'll still be going in that direction, right? So we have this, and Brett talked about drift. You're sort of going in a certain direction. You sort of maintain that direction, right? And you can look at this and it's kind of a weighted effects here. So it's where I was plus, where I was going. And as time gets bigger and bigger, as that delta gets bigger and bigger, you sort of lose this influence, right? Because this term goes away, right? And this term will go away, or sort of keeps me pointed in the same direction because that term goes away and I'm left with kind of just a constant drift. As you get farther out in time, there's less influence. And then we have errors. So instead of that differential equation, we can write it like this. And hopefully you can see now we get this sort of Markov formulation where I'll be in the future, depends on where I was in the past. And that's it, basically, right? And if you want all the gory details of going from one to the other, there's a reference for you. Okay, so let's now keep going in the vein of what we can use. So just like probably everyone in ecology has heard of hidden Markov models, at some point in time, you probably heard about a state space model, right? And basically, a state space model is a continuous version of a hidden Markov model. It has that same sort of form, right? There's an underlying state that goes through that you probably don't see at all. And then from that state, you make an observation that's from that state, based off that state, but you don't see it exactly how it exists, right? So here, just like with the HMM, we have our state. Remember, that's the location and velocity as we go through time. And this is just that same equation we had before. I just piled all those weights into that T matrix, right? But then there's what we do see. So here with the Z, all you can see with that Z matrix, it's constant. And all I'm doing is picking off the mu X and the mu Y. So what we're saying here is what I do see is the actual true locations plus some error around it, right? And so this is where we start to take into account that location error in our movement modeling, right? And the nice thing is that these distribution for these guys right here, they're serially independent through time. They're just, they're correlated within each other, but through time, they're independent. And these guys are independent in time, right? And so we've set up kind of a nice situation. And I'm sure you guys know, well, how am I going to analyze data that comes like as this with the state space model formula, right? Well, that looks messy. That's not the answer you thought you're. So this is the actual likelihood. It's just like the likelihood for the hidden Markov model. You're just sort of progressing through in time, right? The locations, all, everything previous, conditioning on that, plus where I'm going in the future. So you're just marching through in time. But the problem is that you never observe those states, right? Just like with the hidden Markov model, you never observe them. And so what you get is something in actuality, you need to do this and integrate over all those missing states. And that's your true likelihood, right? Not your complete data likelihood. And that just looks impossible. Like, how would you do that? Well, it turns out, of course, just like with the hidden Markov model, with the state space model, there's a nice algorithm that progresses through in time that allows you to do that exactly without ever doing it, essentially. And that's the Kalman filter, of course. And so that, just like a forward algorithm in HMM, allows us to get that likelihood. in a very efficient way. Okay. Now there are of course some requirements here is that we still had everything was linear. Remember, our state is just a matrix multiplication of the next one, and so that we need that for this, just the normal version, and we need those errors to be normal and independent through time. Now, there's all kinds of extensions to the Kalman filter that if you don't have that, but we're going to avoid that right now. That's a different class, a different workshop for that. So now one thing that happens, and I wish I had the reference, I didn't remember it offhand, but what happens with these models is because you're trying to model an unseen state plus measurement error, is that sometimes they can get really difficult to fit sometimes, because sometimes when you're optimizing all those parameters, it can't quite decide, I have a big error and small signal, or a big signal and small error, so you can get issues like that. So sometimes what you might end up doing is a penalized likelihood, and that's just basically adding what you calculate from the Kalman filter, adding on a penalty that keeps your parameters from getting too crazy. And I can show you an example if we decide to do that one versus other stuff later. But basically, if you look at that and just think, if any of you are familiar with MGCV and penalized splines, that's exactly what they're doing there. It's the same situation. So we can fit a likelihood. We go forward and we calculate the likelihood. We can optimize it. And then in the end, we get some parameters. But one of the things, especially for that Brett was showing with the imputation, we don't just want to estimate parameters. We also may want to estimate where was that animal at a time we didn't observe it, right? Because it's continuous time. I can pick any time I want and say, I'd like to know where the animal was at that time. And so we can say we want to get an estimate. So we can get a prediction, an optimal prediction. And for any of you that follow Chris Fleming's stuff, but then that's another continuous model. You're basically Kriegging in time to find out. It's like a spatial prediction, but you're doing it in time. So it's like a one-dimensional prediction in time, what you're doing. So you can get the predictor and the variance for that. And that's analogous to sort of the not the Viterbi algorithm, with the marginal. When you get those state probabilities, no, not the single best one, but the state probability.
Forward backwards.
Yeah, forward back. Yeah, so Kalman smoother is the backward part. So the filter runs forward and the smoother runs back. And so then you can get, for any times you want, you can get an estimate of that state at those times. And it's also assuming all the other stuff that your estimate will be normal, so you can get confidence intervals and all that kind of stuff with it. All right, that's all the messiness of the map that's behind it. Let's actually look at data and fit a model that's that state space model with it now. So the R package that I'll do this with is crawl, but there's other ones since then. What used to be foie gras is now animodum, is another package. I forgot to get a slide for that. It just dawned on me as I was sitting here. That actually fits pretty much the same model as this. They're the same kind of thing. They go about it a little bit different in that they specify the discretization ahead of time and do kind of a discretized version, but it's the same underlying model. And if you want to get it, it's on CRAN now, but Josh and I decided to kind of drop it off CRAN, and we're going to keep it on our R Universe site. So if you ever want it anytime in the future, whenever someone at CRAN tells me it doesn't work anymore, it's just going away, but it'll be there. And we also have a whole GitHub repository of examples if you want to, sort of like the vignette that Brett has for momentum. We have a bunch of examples. They're just in a GitHub repository. Okay, animal pictures, just to make sure we're good. So these are northern fur seal pups. So Brett's mentioned northern fur seals. I used to work with northern fur seals a lot. And these are pups, older pups. You can see they're kind of getting ready for winter migration. Got that kind of cuddly look to them. They are not cuddly, but they're great. So we put on basically before they leave for migration, we're going to put on one of these Argos tags here. And let them go and see what happens to them. That was one of the big questions for our conservation measures. Where do pups go when they leave? So just a bit of biology to keep it real. They basically leave for migration 5 months after they're born, and they're on their own at that point. So it's a pretty quick weaning cycle for these. So once mom goes, gives birth, and keeps foraging and nursing for a while, and then eventually she takes off on her migration, and then the pup is kind of left and says, well, I guess I'll go too at some point, and takes off as well. And so one of the questions is, what do they do when they get out there? Where do they go? So let's just fit these models so we can look at, locations of where they go and things like that. So this is the basic thing, but we'll do it in parallel because we can for each animal here. So here's what the data look like. Looks like just the same stuff Brett was looking. You have DBID is the individual site, is the covariate, the site they were tagged at, sex, male, female, the time of the location, long, lat, and then location class is the class for the Argos error. They give us at least some information that says this is a pretty good estimate or this is a terrible estimate.
But you don't use ellipsoids here, or the ellipses from 2000.
Oh no, yeah, these are old data. Yeah, so you can have the new Argos data has what's called diagnostic data, and they tell you, they give you actually a little more information. They say there's an ellipse of error that it is, and they say the length of the long side is this, the short side is this, and it's angled this way. And you can actually turn that into an error model that you can fit in this model too. I just don't have it here with this data. Yeah, but you can do that as well. So it's the same type of data, but what I'm going to do, since we're going to do this sort of tidyverse style, is basically if you nest these up now, you can see each row is an individual's data. So there are 83 rows, 56 rows in this list of data sets. So it kind of makes it a little tidier. Each individual is their own row. And so this is the whole, all this code is in the stuff. So don't worry about trying to follow through. There's the whole worked out example is in the code for the package for the workshop. So basically the main fitting function is CRW-MLE, and that is going to fit the model. And we have an argument here that tells it, like with the usual R formula, what type of error model we want. So we're just basically telling it there's categorical location classes. And so for each one, we get a different error structure for that. What the data is, time, name, all that kind of, and I'm fixing some parameters here because if we leave them all open, it has a real hard time fitting it because, like I said, it doesn't know where. So we give it at least some. There's pretty good info on the good classes of Argos there about what they are. So we can kind of fix those and let it estimate the rest. Yeah, and the other bits is just other stuff for fitting it. But when we finish with that, we have our fit function, we can actually make a prediction. So location prediction. So it's going to run the backward Kalman smoother on it. And for this example, because I didn't want to have too big of a data set, we're going to do daily predictions. You could do hourly, you could do minute if you wanted, but it's a long deployment, so that won't get big. And then we are just changing this into if you guys work a lot with the SF package. So we can change it to an SF data type. Filter out prediction locations because it also gives you the predictions at the observed locations too. Return it. And I've wrapped this all in, where'd it go? For each, and it's going to do it in parallel. So for each one of these iterations, it's going to farm it out to a core on the computer and do it embarrassingly parallel. Yeah, so this is just a little dplyr stuff to pull out the fit and the spread. Has nothing to do with crawl. And so what we get in our list here is we have the original, but now we have a column that's fit. So these are all the fitted model index. And then we have all the predictions. Notice it's a lot smaller because I just did daily predictions for this. But so each one of these is a spatial SF set of points that has all the predictions. And there they are, all the daily predictions for these pups. And it's pretty amazing. So they start up here and then all the way down. So that's the whole west coast of California down here. They go pretty far for an animal that's only a mammal that's only 5 months old. In there. So that's just, an example of predicting stuff. Now, if you are, I don't know how many are here, if you're a marine mammal person or a marine person or someone who uses Argos data, I should say, we started this new package called Crawl Utils. It does a lot of that sort of stuff you saw in the fitting function. It gives kind of default good values. It's centered on that. It doesn't necessarily have to be Argos data, but it gives sensible defaults and stuff. And it has some other SF functions that are useful for manipulating that data. But I just wanted to point out in red here, it's highly unstable at this point. I make a lot of changes during the day. But if you wanted to take a look at some of the functions, you can get ideas certainly for code or whatnot that you could use to do stuff. All right. Well, that was just a basic, we did a basic Kalman filter, and because we wanted to predict the locations and see what it's there. So let's try something a bit easier now. And this is basically, make sure it's this is what happens. I think since Brett mentioned it, that if you keep adding states, behavioral states, what if we get to 10 states? What if we try to fit an HMM with 100 states? What if we go to 1,000 states? What would that look like? And so what I'm going to do here is it's kind of the same type of thing. What if we just went to a continuous state? Like there's no forage or rest or whatever. It's just a continuous state that changes through time, that changes your movement. So we can do that type of that type of stuff with crawl as well. Is hang on, I'm off sync here with the laptops versus
catch up.
Yeah, no. So what if we don't want those parameters to remain constant over time, just like with the HMM that would switch between behaviors. We can let those parameters vary over time as well. They don't have to remain constant. Still waiting for that. So, but once you make things in continuous time, it gets really hard to do that because you have essentially something you say, well, I could change, I could vary continuously over time. It's hard to work out all the mathematical details of that as is, because all the math that was behind all the state space version of the differential equation. We'll see what happens here. Depends on those parameters being constant. That's how you get all those results. So a way we can kind of sneak around that is to, let's say, well, instead of it being constant, let's say I'm pretty sure I could at least define some sort of discretized sets of times where I'm okay with it being constant in there. So maybe I'm okay with parameters being constant over an hour's worth of time if my deployment's months long or something like that. So if we can pick these times where I'm okay saying this is where the model could change at those times, even though your data are still continuous, so, you know, you can still get predictions at any point in time or anything like that. I'm picking times where the model parameters can change. Then we can define this temporally varying coefficient model. And that's that we get the same sort of process, right? It just marches through in continuous time. But our observations, well, if it's a time we observed the animal, then it's the same as it was before. But if it's one of those, I'll call them knots for a better word, like in time, it's just missing. Like it's a missing observation. And the nice thing about the Kalman filter is like you can filter right over those missing locations. It's sort of a built-in benefit of the Kalman filter. So let's look at this continuous state movement model. And then so instead of those discrete states, let's say for each one of those time knots that we can have a different value of beta or sigma. Those are the two correlated random walk parameters. And that it sort of varies in time with some sort of covariance, you know, we'll make it be like a time series model through time, things like that. So instead of like the Markov switching, we could have it be, you know, just some sort of time series model through time. And so a nice way to do that is we could set it up as a basis function. So essentially think about specifying something in MGCV that's like S of time. Like we can fit a smooth time curve for each of those parameters as a B-spline. So let's look at a harbor seal instead of a fur seal. So instead now over time, our beta parameter has this form with our coefficients. And the sigma has the same sort of linear form with sort of a prior on the coefficients. And we can specify that prior distribution. All I'm doing is specifying this and this in the model. It's just those two normal distributions putting them together as a function. And then we could fit the same sort of CRWMLE, only this time, you'll notice for the movement model, I have it as a B-spline movement model, and that's just sort of a standard R thing, that tilde BS that's in the spline package. And I give it the number of knots I want, things like that, and fit it just the same. And so what it's going to do is fit those movement parameters smoothly through time. Now, I won't kid you, it takes a long time. There's about 100 and some odd parameters in that it optimizes. So it takes a little while, but not a huge amount of time. And so we can pull those parameters off and just sort of for visualization, we could actually take those parameters and cluster them just to see like, what do they look like if we said, if we had to pick two, you know, states out of this thing. What would it look like? So that's more for a visualization thing, but we still have that continuous state. So this is what we get. So this is the correlation in velocity over time. So you see you get the smooth curve over time. And what that is just the persistence of it. So if correlation 0, there's no persistence and it's a random walk, right? And if correlation is 1, you're going to go in a straight line forever. And this is the velocity. So just, you know, think of the random walk sigma, right? As it gets bigger, you make bigger jumps. And that's what this is right here. And so you can see I just colored it. This is just basically naive clustering of that whole thing. So mostly just for visualization, but you can see what happens when the clustering algorithm, it mostly separates the clusters based on the velocity correlation parameter, that beta. And says you're either sort of, there's no correlation. So essentially it's like a random walk, or there's some correlation. And so what it sounds like a lot is sort of, you know, either sort of this foraging state where you're just kind of going all over, or highly correlated straight. And if we plot this on a map, that's kind of what we get. As we get this sort of foraging looking stuff in the blue, and then the more directed travel in the red. So you can do, there are alternatives to an HMM if you wanted to. Now this is kind of right. There's no way to really include covariates in that. Well, yeah, you could try, you could put temporal covariates in that same B-spline statement if you wanted to do that. I've never really tried that. So if anyone wants to give it a go and let me know how it works, let me know. Okay, we'll stop there. Any questions before we?
Oh, do you know what is the difference between crawl and later crawl versions and CTMM?
CTMM, yeah. So CTMM uses a slightly more general movement model. So they tend to use what they call, what is it, Ornstein-Uhlenbeck foraging, OUF, right? And it's a generalization of this. And so basically what it is, now I wish I had put up that stochastic differential equation. So basically the rate of change of the location is just that velocity alone, right? And then the rate of change for the velocity is this Ornstein-Uhlenbeck equation. So what the OUF model is they put an Ornstein-Uhlenbeck equation on the velocity and one on the location as well. So it's a bivariate Ornstein-Uhlenbeck equation essentially. And so if you reduce the parameters to 0 in the location model, you'll end up with this. And they talk about that in the paper, that this model is sort of a special case of their model. So this one, and actually in the second call that we produce, I'll probably put that OUF model. You're just adding essentially 2 more parameters. So it makes it, yeah, a little bit more flexible in fitting those 2 parameters. But yeah, that's the.
I mean, in practice, how do you decide on using crawl versus discrete model? You saw before, is it just when you have these measurement errors, or?
Yeah, so well, the measurement error thing is one thing, because it's hard to you don't have any kind of measurement capability.
And
Momentum.
no, you do crawl.
Yeah, so in the HMMs, it's hard to account for that measurement error because it's based on the your sort of your observation is the location of the animal, and the hidden part is the state. Whereas here, the hidden part is the location of the animal, and your observation is the noisy location of the animal. So it's hard to take into account there. So that's why that gets used a lot for the imputation part of it. So if you have, any kind of measurement error that you'd like to fit all in one, it would have to be more.
That's the thread that we saw before with the data preparation time.
Yeah.
What we saw before was to use this to make the data, to align it and to make it.
Yeah, and so that's going to be in a section, actually, the sampling stuff that Momentum uses inside. But yeah, so that's what they do is they essentially they do the same, like you said, it's just a wrapper. So they're actually calling that same CRWMLE to fit the model and make predictions on the hour or every 6 hours or something like that. And then using that as the data for that.
There was also package AGMB something with hidden Markov models with AT model builder.
It's HMM-TMB.
Yeah.
Oh, that's, yeah. So Tail Nishlo, that's a very general HMM package, like for not necessarily just movement or anything else, just a general HMM package.
in this model, which is available in Hall, is the continuous ARMA process in the position, not in the velocity available?
No,
That would be an exponential covariance.
in the position process, yeah,
Yeah, so that is not.
no. Not yet, hopefully in the next one. Yeah. Okay. So let's take a dive into actually what happens. So Brett showed all these pictures about drawing realizations of these spaghetti plots that then for each one of those imputation tracks gets fed into momentum and whatnot. So let's look at that, how the sausage is made here essentially. And so it's basically what you're drawing. Each one of those tracks is a realization from the posterior distribution of that track, of those locations. So the posterior that we'd be after is this, so we have both our parameters and the missing states given the actual data that we observe. That's the posterior distribution we're after. And so we can factor that out as our usual state space model, right? So here's the state space model. Here's the observation model, the state model, and then you have to have priors because it's Bayesian sense, but and the priors. So that's the classical version. But you can actually rearrange it proportionally to look like something like this. So the conditional distribution of the state given the locations and the parameters, but then times instead of the sort of marginal, the posterior of the parameters themselves. So this makes it, even though it may not look like it at all, makes it a little bit easier to actually come up with something computationally efficient to draw those tracks. And so the key is that we can just draw one of these values from this distribution. And remember, once we fit that, and we have great ways to evaluate the likelihood with the Kalman filter that averages over the state. So we can actually get this or approximate this part relatively easy because we can just evaluate the likelihood. So if we can draw from this distribution, take a draw from here, and then take a draw from this, because given I have the parameter values, there's a really nice algorithm that draws from the posterior of the states as long as you know what the parameter values are. And it's basically the same thing as the Kalman smoother. You go forward and come back, but instead of taking the best value as you go back with the smoother, you just take random draws and you can draw a track, a posterior track from that. And so all we have to do, draw from this, voila, posterior track is in our hand. So that when in Brett's discussion where he's put M equal, forget the argument name, 30 tracks for his MI version, this is all it's doing. Draws one of these, store it. One of these, store it. So that's all it's doing. So for that. And then, so anything you want to do from that track, so any sort of summary of the parameters in a track will be a somewhat summary from that posterior. So you can think of this F, maybe that's fitting an HMM model in there, or maybe it's calculating the distance traveled or anything like that. You get the posterior of that given just the data and you average over it like that. So here's the real messy part of this, is that drawing the states given the parameters it's just like the Kalman smoother. There's an algorithm backwards. But we don't ever have this, you can do all this MCMC if you want, and then you would get an exact sample. But there are several different ways we can do this. And we don't necessarily need to go through all the details of this, but we could do a normal approximation for that parameter posterior. And that's simply because I fit this with maximum likelihood, right? So I'm going to get a maximum likelihood value. I'm going to get a covariance matrix. And we can just say, well, let's pretend that's close to the posterior. Just like with the MLE and the posterior are going to converge at large sample size. Let's just close our eyes and pretend we're at a large sample size. So that's the first one. The second one is you can actually draw an importance sample, right? Because I can evaluate the likelihood. So I can propose a value and check the likelihood and weight it and then do a sample importance free sample type thing and actually get something more exact. Or you could do something exactly kind of like what Inlet does. Let's kind of split the difference maybe between those two and do kind of a deterministic thing. We'll kind of search out on a grid for good values, weight them by the posterior, and then we can just draw from those values, you know, sort of a weighted sample of those probabilities. So you can do any one of those, or I forgot to put on here, you could do nothing and just use the maximum likelihood value and pretend there is no variation. That's another option. And chances are you may find that doing that is the easier approach, because when it comes to all that other stuff, you know, all the variation and what happens on the other side tends to mostly occur because of the location uncertainty or the measurement error. In fact, the variation, the parameters usually has very little, not a lot, but some, but you know, it can be easier to overlook the amount of variation that the parameter leads to in your end result versus the location uncertainty, right? In this, and so that is an option that's not necessarily the worst option that you could think of. So inside the crawl wrap function, essentially this is what's going on. Maybe not the dplyr stuff in there, but this is what he's doing. So once you fit your model, you create a CRW simulator. And this is an object that, you know, it's kind of like creating your own R norm, like so you can draw samples from. So that's what we're doing here. You know, we can tell it, I want a sample location every 6 hours along that track. So I want locations every 6 hours. What method do you want to use? So quadrature is the INLO like method. Quad ask false. So basically starts to evaluate how many quadrature points it needs to assess when it does this. Sometimes, you know, if you have 6 parameters and it has to go a long way in each direction, pick up 6-dimensional space, sometimes you end up evaluating tens of thousands of parameters. So by default, it's true. So it'll say, do you really want to do this? And maybe doing like if it's too much, maybe doing the normal approximation or importance sample a little easier. Like if you wanted to do the importance sample, you could say importance sample here. And then all it does for each time you call that, it's just going to draw one of those tracks out there. And so that's all it's doing there. Each time you call it, it draws a track. And here we have this, we're converting it to a long string SF object just so I can plot it in there. And so this is what we get. This is basically the same tracks that Brett's been showing you, right? So each one of these little red squiggles is one of those 20 sample tracks. So these are all coming from the posterior distribution of that track, given the data, the locations we did observe, even including the location error too, right? Given the noisy data we observed, that's a sample from the posterior. So for each one of those tracks, you can do whatever you want to do. If we zoom into the messy area, you can see where the gaps in time are, and it spreads out. So there's all kinds of, for this section kind of finished here. So there's all kinds of reading for this, that Durbin and Koopman is a great book for just state space modeling. But not, and then the original data, and I wanted to point the original crawl paper. And I wanted to point out this one because it's relatively new. It's Teo basically took that same varying coefficient model. He has a bit of software that expanded it out a little bit so you could do more things, like instead of a smooth through time, if I had my temperature through time, I could do a smooth of temperature on those coefficients instead of just through time. So that's a good paper to read for those that if you want to extend that varying coefficient more than I have here. And he's got some TMB software. So I'm still awake for that. Any questions? And I think we can probably take a break after this. And then we can start making hard decisions about the material encounter. Everyone's asleep. Okay.
Everybody hanging in there?
And the home stretch after this. Okay, guys. Now we'll come down and we'll make the choice. Want to look at any of the population level inference, or just like go faster through this, and we'll walk you through as much code. All the code's available with the package we'll give you for the thing. There won't be as much explanation as it is I go through. So if you want, we can go through this quicker and get through some population inference stuff. I see a few head nods, but okay, we'll do that instead and see. Okay, so this is the next section for continuous time modeling, but it's going to be a little bit different than you probably have thought about before, although we have seen a few examples of it at the conference this week. And that's, we're going to stay with continuous time, but we're now going to go to discrete space. So I'm going to move to discrete cells rather than just completely continuous in space. Okay? And if you remember, Clark Rushing's talk is essentially this, but instead of just states of wintering, migrating, summer around 3 states, our states now could be every cell in a raster. Okay, so think of a whole big raster, and those are all the states. A state is now a location. Okay. So let's look about it now. Why discrete space and continuous time? Well, the question is that as we looked at some of these, right, and I told you can make a prediction with crawl at any time you want. So if I wanted to, I can make predictions every second, every 2 seconds if I wanted. Would that be that useful to you? Well, unless certainly for these animals that we showed you, probably not. Those are big, long deployments, and you don't even believe we know what's going on at the second level anyway, when you're getting locations every hours, few days. It doesn't make any sense to have that. Or would it make sense, if I gave you a prediction a second later from the last time I saw the animal, do you think the animal's done anything interesting in that time? It couldn't even really go anywhere, right? So one of the arguments for the discrete space version is that do we really care or believe we have information at that fine resolution? So we're usually trying to relate use or movement to a habitat grid. That could be, well, this is a little bit old. You'll see we usually get finer rasters for marine layers than 40 kilometers now, but it used to be up to 40 kilometers for the open Pacific Ocean. Why are we trying to relate, this minuscule amount of use when our code area is on a 40 kilometer grid? So rather than trying to model the movement in continuous space, let's bring the movement and describe it in terms of those raster cells. Like, how long did I stay in this raster cell? I moved to that raster cell next. Why did I move there? Is the habitat better or something like that? So let's try to describe the movement relative to the habitat that we have. Or you could also do discrete patchy cells. Like, maybe I don't care what happens in between when the animal is transiting between different areas that I know are hot spots for foraging. I just want to know what happens in that area. And so when did it move from this foraging area to that foraging area? I don't care what happens in between. So those are all sort of different reasons why you might want to go down this road versus the other ones. So let's look at, so this is, it's going to take a minute here. It'll pop up in a minute.
Is that a 30 megabyte slide there?
I don't know. It's not that. I don't think it's. Oh, hang on. I think this computer dropped out of the thing here.
It might be.
I think it dropped off the
Zoom.
Zoom. An unknown error occurred, it said. All
I
right.
get those all the time.
Yeah. It's so just like.
See, where's my? I think I gotta sign back on.
I wonder if Mevin's actually still there.
I don't know.
There he is, there he is. Hey, Mevin.
We need some jokes or something.
Yeah.
Not.
Yeah.
Any jokes?
Well, I,
By the way, I was going to say,
Can't hear
oh, you can't hear me?
you. I'm
Oh,
sure
probably
we can't
it's
hear him.
not
oh,
turned up.
it's- my computer isn't hooking up.
Yours is off? Oh, no.
Not hooking up to the Wi-Fi. We shut our conference Wi-Fi down already?
Oh, I don't know. You can't connect to it?
No,
Denying your.
no, it's just not even connecting.
Are the slides on this one possibly? Are those slides on this machine?
No,
Why don't we download the slides from this machine? If you still have internet.
Yep,
Sure.
yeah, I wonder if they, yeah.
Oh, good point. Yeah.
What's your password?
We have a thumb drive. Stop this. Oh, what is that? No, it's good. I think we have a thumb drive. We can. So the thing's not even coming up.
Oh,
I think the login is for 4 hours and then you have to re-log in.
it's not even hooking up to the router.
It should be until 7 in the evening.
Do we want to go onto your phone?
I don't know if I put no, because I'll run out of juice. I'll run out of,
Another computer, you're welcome to.
They use these sticks maybe. It's easier for you to put the presentation.
Yeah, if you have a flash drive, that would work.
one.
Yeah, the internet's not working.
Share my data from my.
Here, I got,
Oh,
I can put it.
we can put it, we're gonna, I think we'll try. Yeah, sorry, everyone. Technology goes bad.
Now you're gonna have to do that really fast.
I know, I'm just gonna. Or there's some technical bits where we'll skip over.
Up there.
Is that the one I was sharing before?
Can you guys hear me yet?
Yeah.
Yes.
Can you see those slides, Devin? I put them up there.
Oh, yeah.
Oh, thanks.
I was wondering where they were coming from.
You want to just
Yeah,
you
you
wanna
can take
just-
them.
do it that way?
No, you can take them off. We have them.
Oh, okay, okay.
All right. Back in business. Okay. We're back in business here. Can you see them, Mevin? Okay.
Yes, you can see them.
All right. Okay. So this is what we were trying to do, right? Here's our blue dot, here's our observed location. We were trying to make inference about this path going through. Okay, what are we going to do now? Going to do this instead is try to model that path in terms of just the cells, because that's all we care about. So when did I move to a new cell? Why did I move to that cell? How long did I stay in the cell? That type of thing. So that's really the type of data we're looking at before. So instead of this track over here on the left, where we have just this continuous thing, is we're just looking at in terms of modeling the cells I did visit and how long I stayed in the cell. So it actually kind of makes nice sense that I'm trying to model where did I go to and how long did I reside in that position. There are variables that cause me to stay longer there. Maybe I like the habitat, so I'm going to stay longer here as we go. So this is where we'll start catching up here. So let's look at some math behind this. And so this basically is that same continuous time Markov chain that Clark Rushing had talked with before. We just have all kinds of notation to go with that now. So we have cells because we have to worry about, now we have sort of a spatial relationship now, not just, and he had talked about, well, I can't move from summer or winter habitat to summer habitat without going through the migrations to happen. So in terms of movement on a big spatial grid, well, I can't fly or I can't sort of spontaneously transport to another cell without going through other cells. Now we can start, because there's a lot of bird folks here, maybe we can sort of rearrange that, you can sort of say, well, technically I'm in a flying state, but I still got to go through that cell. I'm just doing a different form of movement. But we'll just say you can't transport to another cell. So I got to move through my neighbors to get there. So I have times that I transition. I have a residence time, how long I stay in the cell I'm at. That's just the difference of the transition times. And we have what's called an embedded chain. That's just the list of the list of the cells I visited, no times associated. Just cell 1, cell 2, cell 3. Then went through. And so our whole discrete space continuous time path is that collection of residence times and cells I visited, right? Because that'll any point in time over that deployment, I can figure out where the animal is with those pieces. So I've already told you it's a Markov chain, continuous time Markov chain. So that is that cat's out of the bag. So the process, so basically, where is that, just like Clark had described, where is that animal in terms of a state? Where is that at any given time? We can evaluate that. But we need some sensible rules for how that movement occurs. And one is that looking at a narrower and narrower window of time, that probability of me making a transition has to go to 0, right? Because if I look at a window of time of virtually 0, the chances of me transitioning to the next cell, not very big, right? So it has to go to 0, but it can't go too fast, right? We can't have it go too fast or I won't move anywhere. But one of the real things is if you get a small enough window, I can't make 2 transitions, right? So I can't sort of simultaneously make 2 transitions. Because the time window shrinks down that I'm looking at in terms of a movement. It gets to be a point where it's virtually 0 that I will make 2 transitions in that amount of time at a faster rate than just 1 transition. The last one is a little more technical. It's that the probability of not making a move is probability I don't go to another cell. There's no other option, essentially. That's just this last statement here. So if we have these 3 rules that govern our movement, we can start working. So we have a rate of movement. So that was that lambda, the rate I move to the next cell, and that's how we're going to describe movement, rate at which I move. I think Clark used a Q matrix like the entries. That's the rate that you go to the next state, right? So the next cell. And so we have the probability that I'll move to the next cell over a gap in time. It's just this ratio. So it turns out if given I know I'm going to make a move at a certain time, well, that the cell I move to is kind of easy. It's just the proportional to those rates, right? So it's sort of a multinomial thing. If I say you're going to move in 1 hour, the cell I move to is just going to be a multinomial choice then. And for this, the time it takes me to move that 1 hour, like I said, well, okay, when are you going to move? You'll say, well, I'm going to move in an exponential amount of time. You're going to draw an exponential random variable and then move. So that's basically the list of this model, and we won't go through all that because that's messy. And oops, I think I went backwards. Like going backwards. Okay. Enough math, but that looks like a lot of math. This is statistics now. So if we were to, okay, well, let's fit this model. Well, I already told you the amount of time you're going to reside in a location is an exponential. And then when you do make a jump, it's going to be a multinomial. Right? So we can just, like what you do at coming up with a time series model is you just stack them all together in time, right? So that's what we have is the residence time is an exponential, and then the other section, the cell I move to, is a multinomial. And we can piece all those things together, and we can do on the blackboard crossing out of denominators and numerators that that cancel with each other. And we end up with the second thing right here. And that may not look any better, really what we have. But if you look close, what that is that's just a Poisson likelihood function, Poisson distribution function, where Z is just an indicator of, yes, I moved to that cell at that time, and it's 0 if it's not. And so what you get is just this big product of, it looks like Poisson distributions, even though the data that you observed aren't Poisson in any way. So we get Poisson data with rates R times lambda for that. So we can set up, all right, well, what's our model, our rate for movement? Well, we can set this up where we have the rate at which I move to a cell might depend on two things. It might depend on how good it is where I'm at, or it might depend on how good it is over there, right? So if it's really good here, the rate at which I move will be very low, right? If it's really good over there, then the rate at which I move will be higher, right? And so we can parse those out into two types of covariates. The M's here, the M covariates are what it's like where I'm currently at. If that's good, I'd like to stay. But the Xij is like the relationship between this cell and that cell over there. So if I like that, I may want to move that. So you can model sort of two types of influence, the rate at which I like it and the rate at which I like it where I'm at. And so we have all of those, you know, you can think of gradient type things. And then the best part here, so we've set that all up. That looks like a nice log linear model, but that log linear model drops into that Poisson likelihood. And all of a sudden, this model we can just fit with any GLM software you want. You don't need any special movement modeling software. Basically, the whole aspect of this is just rearranging it to get the data elements you need. And then you can just use GLM, you can use GAM, and MGCV, you can use whatever you want. It's family equal Poisson, length equal log. You can fit these models. So let's actually do something like that. Of course, we're going to go back to these guys right here, our northern fur seal pups. So this is that same animal I showed you guys with the imputation. So let's say we have some covariates that we think might influence how long I'd like to stay in a particular spot or go. So we have, over the course of this deployment, we have sea surface temperature. Maybe I like it warmer, maybe I don't like it warmer. You know, we want that. We also have the surface winds. You know, that's sort of a thing that might be pushing me towards another cell. Do I like to go with it? Do I like to go against it for some reason? So we have this sort of covariate. That's one of those ones that would push me from this cell to that cell right there. Then we also have geostrophic current. Maybe the current carries me to that next cell, modeling movement. Now, of course, I gave you all this information like you actually knew the exact times of transition between the cells, right? And exactly the cell that you transition to, right? But certainly we've already seen with that data that we don't. So we can just follow the same imputation approach this time. And then so we'll fit a track. We'll use crawl to draw that track, just like the same stuff we did before with this animal. And we'll convert those known continuous tracks. Of course, it's kind of discretized to as small as we want to make it, to that function, the times of transition between cells and which cell I transition to, right? So I can turn that to that discrete space continuous time track, get what I call a model data, so the Z's and the spatial covariates and stuff like that. There's a package called CTMC move that fits these models that will extract that stuff for you. But CTMC move doesn't have a fitting function. It's all data manipulation. Why? Because we can fit those models with GLM or GAM or whatever. We don't need a special fitting function. So then we could fit that model, we'll save it. Just like Brett does with the HMM models, we can save those results and then combine them back together in the end. I'll skip through this. So this is basically going, this is the same code for the imputation and stuff like that. It's in the complete examples in the package of stuff. Yeah, so basically this is just going through, you know, forming those discrete paths here, the last line down here, path. And the next line, so I'm just converting that to the discrete space, getting the data I need for the GLM, that CTMC to GLM. My model statement, so I'm going to do sort of a smooth SST, smooth wind, so temporally varying effects of sea surface temperature, wind gradient, current gradient. And then the best part is that the bottom line there, I'm just using GAM from MGCB to fit the model. And then I'll store that fitted model object. And we've already talked about that a lot. So this is essentially the effects you see. This is sort of sea surface temperature. Does it affect the rate at which I want to stay? So that was one of the M utilities. I get to a nice warm spot, do I want to stay here? Or maybe I like cold. And what you can see is there's no real effect on that, but you can see, just in case you did see something where it gets negative here, that means, what that means is if it's negative, the warmer the sea surface temperature is, the lower my rate is, right? So that means I want to stay. So when that becomes negative, I'd like to stay where I'm at higher versions of that covariate. But there's no real evidence for any sort of effect there. And this variable is kind of a straw man one. We already knew it wouldn't affect. But if we looked at the wind, do I like to follow the wind? Well, you know, for a good most of this, it's positive. And so that's saying if the wind is high, the rate at which I go follow that wind vector is high. And so I tend to move with the wind there. If it was negative, it'd be the other one. Like I want to swim against it. But they tend to move with the wind. And no real evidence for geostrophic current either. But positive, it's, you know, generally positive the whole way. So that means I kind of go with it, but it's not, I don't think it's really variable enough, the conditions they experience, to say one thing or another, whether it's sort of a significant thing. The other quick thing I'll mention, and I'm going to go on, let you go. We'll do that next time. A whole bunch of new stuff. This has a bigger description of that model, but I wanted to get to very quickly the the population inference. Okay, this I'll make super short because I know we're already done, so I'm sure you're pretty much spent. So this is a general version, but I've kind of geared this toward population inference. And this is something that's kind of new. Brett keeps trying to convince me to do an R package, so I think we probably will at some point here. But basically, so we've gone through all these techniques where we can build an individual like fit models to an individual data. But how do we say something about either fur seal pups in general or bearded seals in general, something about their population? Do this. So there's basically, the first thing someone would say, oh, is make the parameter individual random effects, right? That's kind of the stat solution for doing this. And then we could talk about, the mean of the random effects, the population mean, the population variance, or anything like that. The problem is to fit that all in one thing, you have to stack all that data together, right? And simultaneously fit all those models together, right? So now if you have a really complicated HMM and you have 100 individuals with long deployments, that's going to be quite an ask to just try and get that. So we have that. The thing is, we want to get to that type of model and do it computationally efficiently. So this is what we do. This is the usual thing we do, is stack that all together. That's the random effect model. But the goal is that we'd like to do here is make it a little more computationally efficient, so that we're going to parse out the computation. So you've already done the work to fit each individual model, right? Why would you want to waste that effort, right? So you can see in stage 1 of this, we're going to fit each individual model. And then in stage 2, just like with the multiple imputation, we're going to combine it back together as if we had fit that full model. And it basically boils down to doing, I won't go through all the steps of why, but it basically boils down to, it's a meta-analysis. So I can take those parameters that I've estimated for each individual and just fit a linear mixed model where the estimated parameter is the response. And I have an individual random effect vector. That's the random effect here. And the epsilon is an error where the variance is known. That's the covariance matrix that I estimated from each individual model right there. So when you do that, so here's just a quick example I'm going to do with the discrete space movement model. It was fit to those false killer whale data that I shot right by. So we fit one of these models where we have, space covariance and things like that, and we create a utilization distribution from that. It has a closed form. And so we can fit that model, calculate the UD. Well, now I got, 50 individual UDs. What does that mean? In terms of where these animals are. But we can sort of combine these together using that same method, or using that linear mixed model method. And so we'll do that, we'll fit it, and then so we're going to get, well, there's clusters here, but if there was just one population, we'd get like a population mean UD, and we can get individual UDs that are sort of borrowed strength from all the other animals to kind of shrink them back towards that, the same thing that random effects do. So what we get from all these 50 animals we have, here's the individual population UDs we get from it. But here's all those individual UDs that we fit, right? You can see for some clusters, like the bottom row 5, there's not much information, so they all pretty much have shrunk back to that population mean, right? Whereas like for 3 here, you'll see this one far at the left-hand side for group 3 on the second row up. That guy's a little different. He had a little bit more information to estimate that UD versus another one. So it didn't get shrunk back all the way. So basically, in a nutshell, that's what it is. And if we actually can extend this a little further, if we had all kinds of other parameters, I won't go through the whole thing now, but you could also use this method to fit IPMs. You got 2 different data sets you want to combine together. We won't go through that, but doubly so for this first topic here. I glossed over a lot of stuff, but the thing is that it's weird. It boils down to a very simple thing. Just do a linear mixed model of the parameters is what it boils down to. Yeah, and whatever software you want, fit all your animals in HMM or crawl or whatever, or stan or nimble, if you want to do it as a Bayesian. You can do that too, and it's a bear, like every animal can be fit in parallel, right? And then combine them together. Yeah, and if you do it in stan or nimble, the paper that Mevin actually quickly flashed up there is a more exact way to do it. Like you can actually physically combine those MCMC samples together to estimate those random effects. So this is kind of the approximate version to do that. So with that, more information if you want it. Again, it's on the stuff we'll give you. And it was a great Euring conference. Any other quick, because I know I just blasted right through that. So stuff is out there for population inputs. We just have to get it more out in the open.
Thank you. Yeah, it's cool that you guys are thinking about that.
I was trying to get Mevin up there again. Yeah. Okay, putting Devin back up there. Thanks for, well, everyone, thanks for hanging out for the full workshop. And Mevin, thank you.
Material.
Plus bonus material, yeah. So yeah, unless there's any other questions, I think, if there's interest, Devin and I could still hang out, or we could go to a pub or something. Otherwise, yeah, thanks for coming and hanging out with us here on the last afternoon of the last day.
Anything, Mevin?
No, I thought you guys did a great job, and it's great to see everybody. Sorry I couldn't be there. Next time.
All right, see you later. It's June sometime, probably, Mevin. You're in Fort Collins.